Hello and welcome to Apologetics 101. I am your host, Christian apologist, Brian Keith Bowen. All right. Today we are interviewing uh, Donny Badinsky of Standing for Truth Ministries. And we're going to be interviewing him on the subject matter of did chromosome 2 fusion happen? This is where evolutionists claim that two different chromosomes in chimpanzees, which they call chromosome 2A and 2B, which I think is like chromosome 13 and 14, but they renamed it 2A, 2B because I think that they fused together in order to justify why humans have 46 chromosomes and chimpanzees have uh, 48. They think that two of the chimpanzees' chromosomes had fused together to make what is known as chromosome 2. And so there's a particular site that they consider to be the fusion site. And we're going to be discussing that along with the alleged chromosome 2 fusion. We're going to do a little bit on ERVs. And Donnie's going to be bringing a presentation now. And we are starting right now. I'm here today with Donny. How do you pronounce your last name, Donny? Badinsky?
Badinsky.
Badinsky. Okay. Donny Badinsky from Standing for Truth Ministries. And he's going to be giving a presentation on chromosome 2, the alleged chromosome 2 fusion believed by evolutionists and in ERVs, right? Is that
That's
correct?
right.
All right.
Yeah.
And he's going to give a presentation, and then we're going to ask some questions afterwards. But Donnie, could you introduce yourself first and your background and everything, and then you can jump right into your presentation if you want.
Absolutely. Ryan, firstly, you are doing some great work on your channel here at Apologetics 101. I appreciate it, brother. You're standing for the truth, and my ministry is called Standing for Truth Ministries. And so I've been doing this now for about 4 years. About 4 to 5 years. Time flies by, put it that way, Brian. And I've got a team. So there's a team of us that put out content. We do a lot of research behind the scenes. As you know, we host a lot of debates. And so I've hosted now over 320 debates on all sorts of topics. Creation evolution is one of the main topics, of course. I engage in that topic as well. We do a lot of open mics. And we also host a lot of debates on theology, soteriology, nature of God, eschatology. And so my ministry, it puts out a lot of content on a lot of different subjects, but my main area of focus is genetics and the topic of ancestry. And so I've written numerous books. Two of the books that I've written, two of the more prominent ones is the Endogenous Retrovirus Handbook. I'll briefly be touching on ERVs tonight, but I'll mostly be focusing on the chromosome 2 fusion argument. And in my other book titled Special Creation, I have an entire section in that book on the chromosome 2. And so I'll be focusing on a lot of that content. So again, Brian, thank you for hosting this, brother. And you keep up the good work.
Awesome. All right. Well, whenever you're ready for your presentation.
Okay, cool. Okay. So I guess I'll just screen share. So it's coming in full screen. Looks good.
Yeah.
Okay. All right, let's get started. I'm looking forward to this. Brian, again, thank you for inviting me on to your program. Okay, so debunking evidence for common descent, chromosome 2 fusion, and endogenous retroviruses. So I'll mainly be focusing on the chromosome 2 tonight, but I will touch a little bit on endogenous retroviruses because in being somebody who's hosted so many debates, I've engaged in so many debates, I have come to realize that two of your main lines of evidence often utilized by the evolutionists is chromosome 2 fusion and endogenous retroviruses. If you look up or do some research, what's the best evidence for evolution, typically ERVs comes up, and then also chromosome 2. And so that's why I've chosen to focus a lot of my time and effort on addressing those two main arguments, since so many evolutionists in the evolutionary community and the debate world look to those lines of evidence as basically irrefutable arguments for common descent. And so we're going to debunk chromosome 2 comprehensively, and then I will also briefly touch on endogenous retroviruses. So this presentation will be presented by myself, Donny Badinsky, founder of Standing for Truth Ministries, my website down there, standingfortruthministries.com. When it comes to the chromosome 2, I have specifically engaged many of the critics on the topic. I have, overall in debates, I've done 100 formal debates, but I've done a lot of open mic and impromptu discussions and debates as well. And so if I were to account for those, I'd probably be between 200 and 300. And so I've got a lot of experience confronting the critics and just having back and forth engagement with them. And so here's a few specifically on the chromosome 2 fusion. And so I've had a formal podcast debate on the fusion. It was about 3 hours, so it was thorough. Grayson and myself, Grayson, he debates frequently as an evolutionist in the debate community. I've also done in an open mic, I've engaged both Mark Reed and Grayson on the chromosome 2, and I've also engaged Dr. Dan Stern-Cardinal on the chromosome 2 fusion. And you can check all those out online and also on my YouTube channel. I've written 2 extensive articles on the topic. You can find them on my website, standingfortruthministries.com. The top one there, overturning the human chromosome 2 fusion, you can find that as an appendix in my special creation book. And then the other article available on my website, refuting Gutsick Gibbon on the human chromosome 2. And so that article focuses a lot on the work that Christopher Roop and I have done. Chris Roop is the author of Contested Bones, fantastic book. I highly recommend it. It does an amazing job just destroying the so-called hominin fossil record in human evolution. And so we discovered, Chris specifically ran the BLAST analysis, we discovered many sequences of these, and I'll get into it in my presentation, your TTA, GGG, CCC, TAA, your telomeric repeats in both forwards and in reverse. We've discovered them in mirror form. And in the chromosome 9, we found an example that is pristine, a lot more pristine than the so-called chromosome 2 fusion example. And so that article deals with that, showing the critics that we find many instances in the genome of these repeats in mirror form, back-to-back organization. We'll get into that during the presentation. Here's a couple of the books I've written: Special Creation, Dismantling Evolution and Confirming Independent Origins. In this book, I specifically focus on the genetic data confirming separate ancestry. So mitochondrial Eve, Y chromosome Noah. And I've also got a specific section on both the chromosome 2 and also endogenous retroviruses. The other book that I've written, the Endogenous Retrovirus Handbook, this strictly focuses on ERVs and refutes all the most common arguments associated with ERVs or ERVs, and also answers the most common questions like their nested hierarchical distribution, answers the questions surrounding the mutations found in the ERV sequences themselves, and why ERVs look the way they do. We'll get into that a little bit in this presentation. And I've also written a comprehensive book on eschatology, End Times Revealed, Dawn of the Antichrist. Here's my website, again, standingfortruthministries.com. If you check it out, I've got an extensive list of videos and just questions on the main page. If you scroll down on the main page, look to the right, I've got dozens of common creation questions that you can click, and it'll either take you to an article we wrote or a book or a video or even a debate. So there's a lot of resources there for people to check out. Okay, so before we get into the really technical aspects of tonight's presentation. I do want to be thorough, and I do want to break it down, though, in an easy to understand way, as a lot of these genetics topics can be technical. And so I want to start off with a couple mottos that I believe we can all learn from and live by. And so the first one, believe God more and believe man less. I first heard this from Dr. John Sanford, the writer of Genetic Entropy. Excellent book, major game, life changer, changed my life. And so believe God more and believe man less. The Bible talks about how we shouldn't put our faith in the wisdom of man, but in the power of God. And that is so true. Good science always affirms Scripture. The Bible claims to be the history book of the universe. And so we should be able to look at the world around us, and we should be able to look at the empirical scientific evidence and test it to the claims of Scripture. And Genesis specifically gives us an account of our origins, the origins of the universe, the origins of the earth, the origins of man, goes into the global flood, the Babel dispersal, and all scientific data affirms those events. And so I think that's amazing. So here are three of what the evolutionary community believes or asserts are the best lines of evidence for common descent: endogenous retroviruses, chromosome 2 fusion, pseudogenes. What's funny is a lot of these arguments go back to the junk DNA paradigm. When you overturn or refute junk DNA, which we understand that most of the genome has amazing evidence for activity, the ENCODE project discovered that over 80% of the genome has biochemical activity. A lot of the evolutionists will say, oh, this is spurious or this is just noise. But adding just a single nucleotide to a growing RNA change consumes energy. It consumes ATP. And so if this really was just junk or spurious, then processes like natural selection, if evolution was true, should have weeded out all of this genetic baggage over time, as it would be wasteful of energy and resources to transcribe and replicate this DNA data, basically. And so within those regions that are active, we have pseudogenes. There's many papers that I could go over that talk about how they are necessary to sustain healthy life processes. Endogenous retroviruses we're going to get into during this presentation, and the chromosome 2. The two main arguments that I will be focusing on, ERVs and chromosome 2, but with a real strong focus, detailed focus on the chromosome 2 fusion. Before I get into the chromosome 2 fusion itself, for the audience, I want to go over some important terms and just briefly cover the different models here. So we got the separate ancestry model. That would be the model that I hold to, that Brian holds to, that essentially says man was created separate and independent from any other form of life. As you can see on the diagram here, on the visual, we have humans going back to just two common ancestors, Adam and Eve, in the not so distant past. You have some extinct groups like Neanderthals, Naledi, Hobbit, more technically referred to as Homo floresiensis, and then we've got living humans today, us. But as you can see, humans are not connected to any of your great apes, orangutan, chimpanzee, or Australopithecines, for example. And so we would argue that humans are created separate and exist separately from every other form of life, including the chimpanzees. Evolution, on the other hand, would argue, evolution by common descent, that is, would argue, as you can see here on the chart, this tree of life, all life, including fish, amphibians, marsupials, birds, humans, crustaceans, all go back to a single common ancestor. And so they would argue that all plants and all animals of all time are related to a single common ancestor. Again, here's an example, the tree of life, all life, including strawberries and whales, going back to a common ancestor. This is a more sophisticated phylogenetic tree, but it's saying the same thing. Bacteria, birds, mammals, and reptiles, to the evolutionists, all go back to a single common ancestor billions of years ago. So one important thing that I want to point out in any debate on evolution, we need to define terms. And so you'll hear the word evolution. And critics will say, you don't believe in evolution? Well, it depends what you mean by evolution. So if by evolution you mean change over time, then sure, nobody argues against change over time. Cell phones change over time, cars change over time, we change over time, our thinking processes change over time. Change over time is not disputed. You can even say that all dogs, let's say all domestic dogs, go back to a single common ancestor, and that would be a wolf-like ancestor. So if you were to argue that wolves, coyotes, dingoes, domestic dogs are all related through common ancestry, go back to a single archetype 4,500 years ago, we're not going to argue against that. Yeah, change in allele frequencies, allele being a genetic variant, and the frequency of those variants in populations over time, sure. Nobody argues against that. But if by evolution you mean dogs, wolves, and banana plants are related through common ancestry, that's where we as biblical creationists have a problem. And so the evolutionists will look to time as the hero of the plot. With lots of time and natural selection and mutations, right? The driving force of evolutionary change to the evolutionists is mutations. Apparently, create the novel genetic variation that natural selection can act upon. And over time, these microevolutionary variations can lead to large scale or macroevolutionary changes. But the problem is mutations are overwhelmingly deleterious. The vast majority of mutations are nearly neutral, meaning they're only slightly deleterious, like a single rust molecule on a car, or a single spelling mistake in a book the size of an encyclopedia. On their own, the effects are inconsequential. But as they build up over time, the car rusts out and degrades, or a book degrades. The message is lost over time. And that's what we see in the genomes of biological organisms. Natural selection is just a fine-tuning mechanism. Keeps a species as strong as it can be. And so more time actually means more deleterious mutation accumulation. Natural selection can't see most of these mutations, so they build up and they lead to genetic sickness. And therefore, no, more time is not going to take your fish to fishermen. More time is not going to take your bacteria-like organism to biologists over time. More time means extinction, more degeneration, and no forward evolution. Okay, so let's get right into the main topic, chromosome 2. I'm looking forward to this. So since protagonists of evolution believe the great apes, now evolutionist taxonomists, they would include humans in the great ape family, according to their model. And so they argue that all the great apes, including humans, according to them, must share a common ancestor. And they have appealed to an imaginary fusion event in the human line at a point in time after this hypothetical split between the human and chimpanzee common ancestor. They'll say that this split occurred roughly 6 to 10 million years ago. And so you've got humans today as an extant species, chimpanzees today as an extant species, and both species go back to a common ancestor. They go back to a point roughly 6 to 10 million years ago. They argue that the fusion event would have occurred after the split in the human line at some point. Okay, so as you can see on the visual here, apes have 48 chromosomes, and humans, whom evolutionary scientists classify in a subgroup of primates known as the great apes, have 46 chromosomes. Humans stand out as the only species within the great apes as having 46 chromosomes. The question is, why the difference? Well, to the evolutionists, there has to be an explanation or a reason behind the difference, because they assume universal common ancestry. They assume that chimpanzees are our closest living relatives and therefore go back to a common ancestor, humans and chimpanzees. And so they propose that there must have been a fusion event. And they would argue that this is the most parsimonious explanation rather than other explanations that they could look to. And so they'll argue again that at some point after the split, there was this unilateral mistake that occurred, resulting in humans having 46 chromosomes rather than 48. And then so now you have your other great apes, 48, humans have 46. Then you would need this fusion, this unilateral mistake. Once it arises, it would then have to spread and become fixed. Fixed just means to be stuck in place, as in every single human being today would have to have 46. So that change, that mistake genomically has to be fixed where everybody has it. Every single human has 46 chromosomes. Nobody has 48. Okay. And so defenders of common descent, they assume that humans and chimpanzees are related to a common ancestor, and therefore they expect to find evidence for this fusion somewhere in the genome. Here's a diagram going over what would be expected if there was a fusion. So here you've got chimpanzee chromosomes 2A and 2B. Okay, your end caps of chromosomes are telomeres, telomeric DNA. Over time, they break down. That telomeric DNA is lost, basically. It withers, much like the end caps of a shoelace. You got that little plastic piece at the end of your shoelace, and it protects the shoelace from flaring out and degrading. Well, over time, usually, if you're familiar with your shoes, that plastic piece will degrade. It'll break off, and then your shoelaces flare out, and you need a new pair. And so that's akin to how telomeres work. They're the end caps of chromosomes. They protect the chromosomes, as a matter of fact, from fusions. And so if there is a fusion, you'd have these two ape-like chromosomes fused together, where in the middle, you've got your telomere DNA. But you'd also have a second deactivated centromere. Okay, and so here you can see in the hypothetical fusion, you'd have two centromeres. One is deactivated. You'd have your telomeric DNA in the middle, because that's where the fusion took place, the connection took place. And then you'd also have, as usual, your end cap telomeric DNA. And so this is what we should find. The question is this what we have discovered? And we're going to cover that in this presentation. Now, the earliest hint of a chromosome fusion in the human line after this hypothetical split took place was actually prior to genetic sequencing. Scientists were looking at images of human and chimpanzee chromosomes through an impressive technique or process called karyotyping. And in this method, what you can do is basically isolate cells as they're dividing during mitosis. And the staining that actually takes place during this procedure gives a remarkable visualizing of the chromosomes. And what it does is it generates these bands within the chromosomes themselves. And so based on this procedure, based on this practice of visualizing chromosomes and their banding patterns, evolutionary scientists allege that two chromosomes, now actually called 2A and 2B in chimpanzees, that's how confident they are that this happened, fused to form human chromosome 2. For the evolutionists, this accounts for the disparities in chromosome numbers between humans and the great apes. And so why the difference? Well, according to the evolutionary community, this is the explanation for the difference. Now, these images, through karyotyping, that is, are what first gave the evolutionary community the notion or idea that there may have been a fusion in the human line. And advocates of common ancestry, they require this fusion to have taken place. Dr. Ken Miller, a prominent evolutionist, he argued in a lecture, he said if there was no fusion, there was no human evolution. And so if we can falsify the fusion, that at the same time falsifies evolution, shows that it was highly unlikely, even in light of whatever other explanations they could invoke, like fission and other explanations that are just even worse than this explanation. And so if there was no fusion, then they would be obliged to basically invoke another but very convoluted and non-parsimonious answer to why the great apes and humans have differing numbers of chromosomes. Anyways, when genetic sequencing technologies began to expand and become more elaborate, scientists discovered what they believe was the setting of a putative fusion in the human line. That's what we're looking at here. This is the so-called fusion site. Now, as I talked about earlier, there exists a specific type of DNA sequence at the ends of chromosomes called telomeric DNA. Now, in humans, these sequences are approximately 5,000 to 15,000 bases long. Let me repeat that. 5,000 to 15,000 bases long. That's a significant amount, because if two telomeres fuse to each other, even if they were degraded when they fused, we would anticipate a sizable DNA signature or mark, somewhere between 10,000 to 30,000 bases in length. Or maybe, as the critics say, these telomeres degenerated in the first place in order that they can be fused, since telomeres protect from fusion. We're still looking at something in the thousands, not the 798 base pairs that we find in the so-called fusion site, because that's what we uncovered. And the telomeric sequences found at this site, as you can see in the image, they are in both forwards and in reverse. Okay, we understand that DNA is double-stranded. Therefore, your forward sequence is the TTA-GGG, and then that would go on as a tandem repeat, TTA-GGG, TTA-GGG, especially in healthy telomeres. The complement to that, the reverse complement, is CCC-TAA. Problem is, this so-called fusion site right here, it's exceptionally degenerate, and this comes from the conventional scientific literature. This comes from their own literature. They asked the question, why is this area so degenerate? As a matter of fact, the site is only about 70% similar to what a pure fusion would look like if it were 798 base pairs. And so this is very small and corrupt. As you can see here on my cursor, you've got a repeat here, TTA-GGG. You've got one sequence. And it's not even repeated. It's in isolation. You don't get another repeat, or you don't get another telomeric sequence until way down here. Highlight TTAG. So they're isolated. They're only found in a few. There's about 10 here. There's only one that's in tandem repeat where they're touching. TTA-GGG, TTA-GGG. The site that Chris Roop and I found in chromosome 9's much more pristine. You actually get the TTA-GGG multiple times in a row, touching in a head-to-head organizational fashion. But here in the so-called fusion site, they're mostly isolated. They're not connected. They're broken down. It's degenerate. The CCC-TAA, the reverse complement sequence, same thing. Most of them are disconnected. Mostly what we see here is degeneration. You get about 43, if I'm not mistaken. You can count them up yourself. You get about 43 CCC-TAAs. You get about 10 TTA-GGGs. So not very many. It's quite pathetic if it were a fusion. Now, before I get into the details on why the evidence best suggests it probably wasn't a fusion, I do want to answer a few important questions, because in the creation versus evolution debate, Brian, you've probably heard this before from other creation scientists too, is there's a lot of overlap. There's a lot of what's called agnostic lines of evidence. What that means is both models, creation and evolution, or separate and universal ancestry, can explain the data. It would be like me saying creation's true because the sky is blue. But the evolutionists would say, well, wait a minute. Evolution can also explain why the sky is blue. And so it's not discriminatory evidence. It's not a way that can help differentiate the models. If I said, you know what, creation's true because the earth is a sphere, well, evolution can also explain that data. There's many lines of evidence that's agnostic. There's overlap. For example, homology. Evolutionists would say that these homologous patterns are there. Shared structures in the biological world are there because they've been inherited. They've been inherited from past common ancestors. Creationists would say human engineers design in homologous patterns. And so we would expect homologous patterns in the biological world. That makes sense. And so that's an example of an agnostic line of evidence. It's not a way to discriminate between the models. There's overlap. Both models can explain it. So what about the fusion? Is the fusion a discriminatory line of evidence, a way to answer this question of ancestry? Well, here's the questions I want to answer. Was there a chromosome fusion in the human line? We'll cover that. If there was a fusion in the past, which model best explains it? What is the evidence against a chromosome fusion in humans? And that's what we're going to get into mostly in this presentation, number 3. But the first 2 questions, I want to point out that even if, for sake of argument, there was a fusion, just give it to the evolutionists before we get into the reasons why there probably wasn't. Let's just say, for sake of argument, there is a fusion. Is that a line of evidence that only evolution can explain? Well, the answer is no, because if humans had a fusion event, it would actually be a near extinction event. That one person who experienced the fusion would be the ancestors of all humans today. This would rapidly drive a species into near extinction. It would be extremely difficult for this unilateral mistake, this fusion event, to reach fixation. And what you'll oftentimes hear from the evolutionary community, from evolutionary scientists, is whoever experienced that fusion, that lineage basically, where it'd be passed on to, be fixed, all other lineages on the earth, wherever they exist, they'd have to be wiped out. They'd have to go extinct. And so all that's left is the lineage with the 46 chromosomes. Problem is, historically, there hasn't been what's called random mating. Usually humans have come together in tribes, they've come together in communities, and they've intermingled and interbred, reproduced within those communities. And so you have many different lineages, right? And so it'd be very difficult for this fusion to be fixed when you have so many separate tribes on the globe, or even if they're just situated in Africa, but in different parts of Africa. They're going to have to invoke a lot of storytelling, which they have. And I've debated a lot of evolutionists who simply admit that, yes, we don't have really a good explanation for how it became fixed. But the creation model, the biblical creations, we actually have 2 bottlenecks. We have a bottleneck after the flood with 8 people, Noah, his wife, their 3 sons, Shem, Ham, and Japheth, and then their 3 wives. And so if there is a fusion event either before the flood that went through the ark, or sometime after the flood, you have this bottleneck. Then you also have the bottleneck at the Tower of Babel before people spread out in all parts of the globe. And so that unilateral mistake could easily be fixed. Remember, fusions do happen in nature, but they are manifestations of genetic entropy. They are mistakes. They are evidence of degeneration. And so it's not evidence of forward evolution. And so if any model can explain the data, it would be the creation model. Because again, we have the 2 bottlenecks where that fusion could actually be passed on to all people, especially at the Tower of Babel. People spread out to all parts of the globe, explaining why everybody has 46 chromosomes. That is just for sake of argument. Chances are there wasn't a fusion, as we're going to cover here. But even if there was, the evolution model doesn't even explain it as well as the creation would. Anyways, as you can see on the slide here, if there was a fusion, it is what's called an apomorphy, since it is a trait observed in a single lineage. So basically, an apomorphy is a novel evolutionary trait that is unique to a particular species and all its descendants. And why the fusion is defined as that is because this would be a feature that only the human lineage has and has experienced. So it's unique to that particular species, being humans. Okay, well, the creationists can say that it happened in the human line since it's a human specific fusion event. It's not like it happened before the split. According to the evolutionists, it happened after the split. And so we have the evolutionists, the evolutionary model as humans and chimpanzees going back to a common ancestor. Then there's a divergence event. And then in one line, you get a fusion. Well, the creationists would have 2 separate lines, chimpanzees and humans, and somewhere down the human line, you get a fusion. So it really is agnostic to the overall debate. But another supporting line of evidence for why the creation model would best explain a fusion if it occurred has to do with chimpanzee, gorilla, and human chromosomes. And so here's a prominent paper that I have gone over many times in debates and presentations. It's called the Ventura paper, essentially, the evolution of African great ape subtelomeric heterochromatin and the fusion of human chromosome 2. Well, Your chimpanzee, gorilla, and human chromosomes, they actually differ by the presence of what's called large blocks of subterminal heterochromatin. So basically these repetitive segments of DNA that comprise satellite sequences. And they do not exist in human chromosomes, but they do exist in chimpanzee and gorilla chromosomes, telling us that, sure, maybe there is a human-specific chromosome 2 fusion, and we actually have this very specific type of DNA in chimpanzees and gorillas that humans don't have. But if we were related to chimpanzees and there was a fusion event in the past, we should find this DNA, but we don't. And so the evolutionary community basically has to argue that it's been lost in the human line over time, and that's mostly a story. Chris Roop and Dr. John Sanford point this out in their book, Contested Bones. And so here on the slide, we see chimps and other apes have very distinctive and very large satellite sequences at the end of their chromosomes. They are found just before the terminal telomeric repeats. These are typically made up of a very prominent 32 base tandem repeating sequence. These large subtelomeric repeat structures are in all ape genomes, but are absent in man. And so if you're following me here, we have the human genome, human chromosomes. They lack this type of satellite DNA. It doesn't exist. It's not found in the human genome, but yet it's found in chimps and gorillas. But wait a minute, there is a chromosome 2 fusion and we split from a chimpanzee common ancestor. Humans and chimps, that is, go back to a common ancestor in the distant past. We should find this type of satellite DNA around human chromosomes, including chromosome 2, if there is a chromosome 2 fusion. And so, again, for sake of argument, if you wanted to just give the evolutionary community the human chromosome 2 fusion argument, just say it happened, for argument's sake. The creation model far better explains how that unilateral mistake could have been fixed. Okay, so let's move on to the reasons suggesting that it's not a fusion that we're looking at. But either way, it doesn't help the evolutionary model, which is the important point to take from the last section. Degeneration at the fusion site. So I talked about this earlier. Here's a paper that I highly recommend people read. It's very detailed. It's a detailed analysis. So they asked the question, if the fusion occurred within the telomeric repeat arrays less than 6 million years ago, why are the arrays at the fusion site so degenerate? So also to address some criticisms, Gutsick Gibbon put out a video recently, and she tries to argue, like many critics, they argue that the degeneration is expected. The degeneration should be there, or else there wouldn't have been a fusion in the first place. Then why are prominent scientists who were studying this topic back in 2002, why are they surprised? Why didn't they say, oh, wow, we found the degeneration at this site, and that's expected. That's what we predicted. No, the gold standard of science is making testable and falsifiable predictions. They did not predict that the fusion site should be so degenerate. This is post hoc, ad hoc rationalization that we see from people like Gutsick Gibbon. And they can post hoc, ad hoc all day, but it doesn't work scientifically and logically, okay? Because these scientists are asking, why are the arrays at the fusion site so degenerate? Now they go on. Erika tries to say that I quote mine. No, if you watch any of my debates or presentations on this, I point out that yes, if you keep reading the paper, they give what they believe is a few possible reasons for the degeneration. But those reasons essentially amount to storytelling. We're dealing with historical science here. This fusion supposedly occurred in the distant past, millions of years ago. And now they're invoking just so stories to explain away that which they didn't predict. So here are human chromosomes and chimpanzee chromosomes. If you actually do a search for the so-called fusion site in the human genome, you'll find many instances of these telomeric repeats all throughout the genome. And you can see here, chromosome 2, you have many of these interstitial telomeric repeats, they're called TTA, GGG, CCC, TAA. You find quite a few in 21, 22. Nobody would say that these are fusions. Chromosome 16. And so you can find these internal telomeric repeats all throughout the genome, and they have been shown to actually be functional DNA units, important to processes in the cell. And Dr. Jeff Tompkins, he's written a paper on this, and he's shown that literally thousands of telomeric repeats across the genome that we see here have been directly associated with hallmarks of gene expression. The same type of transcription factor binding and gene activity happening at the alleged fusion site are occurring genome-wide at numerous other interstitial telomeric repeats. And so what we're looking at is design functional code within human genes. Now, the critics will say, oh, but the chromosome 2 area has the highest density of these repeats in both forwards and in reverse. Gutsick Gibbon, she put out a challenge. I want you to take the exact fusion site, 798 base pairs, and find it anywhere else in the genome. That's called circular reasoning. That is a logical fallacy. Let's assume that this 798 base pair region is a fusion, the ancient remnants of a fusion. Now show me that exact site down to the last DNA letter anywhere else in the genome. No, that's circular reasoning. What we do find is other instances within the genome. Chris Roop and I specifically found the TTA, GGG, CCC, TAA in both forwards and in reverse, obviously your reverse complement, in a mirror image in that organizational pattern, and it's more pristine. And then Gutsick Gibbon wants to say, oh, well, the fact that it's more pristine tells us it's not a fusion, since it shouldn't be that pristine. But as we showed earlier, they didn't expect that degeneration. So this is a lot of post hoc, ad hoc rationalization. So yes, we do find many instances of the internal telomeric repeats in the genome on other chromosomes. We also find them in both forwards and in reverse. We find them in that pattern in many spots. And we find a very pristine site in the chromosome 9 that specifically Chris Roop and I discovered. That covers all the bases. Now, the density aspect of it, the critics want to say, well, the chromosome 2 area is the most dense, and so therefore it must be a fusion. But again, dense compared to what? That's arbitrary. That's subjective. That's like saying, well, this universe looks old. But wait a minute, we don't have another universe. We don't have Earth 2. Like in comic book world, we don't have another universe to compare this universe with. This is our only universe. And we can argue that it looks young based on a number of lines of evidence. We only have one human genome of 46 chromosomes, one human species. We don't have Earth 2 and Earth 3 and Earth 4, where we have different human genomes to compare this human genome to. And so dense compared to what? It's already far too degenerate, as evolutionary scientists studying this have pointed out. And so people like Erika are guilty of logical fallacies in their reasoning here. Circular reasoning to expect us to find this exact signature elsewhere in the genome. Well, we do find this signature, just not in as high density in the chromosome 2. But the question is, why is the chromosome 2 so dense? Is it dense for functional purposes? They want to say it's dense because it's the ancient remnants of a fusion. But again, that's subjective. What are you comparing that to? We only have one human genome. We have one universe. Don't tell me that this universe looks old. This is the only universe we have. We don't have another universe to compare it to. But notice when you actually look for these internal telomeric sequences in the chimpanzee genome, notice here, you don't find any. You find a couple here and there, but they're generally absent in chimpanzees. And so we could look to many lines of evidence that actually distinguishes or differentiates the models. The human genome has many instances of these internal telomeric repeats. We know they're there for functional purposes like involved in gene expression. We also know that the human genome doesn't have this type of satellite DNA. It's not found, but it is found in chimpanzees. We could get into Y chromosome dissimilarity. We could get into differences within gene expression, gene regulation, epigenetics, alternative splicing. There are so many differences that we could look to between humans and chimpanzees that actually differentiate between the models, that show the separate ancestry model best explains the data. Okay, so let's move on here. As you can tell, human chromosomes 2, 16, 21, and 22, they're peppered with the fusion site sequence over the length of their entire euchromatic landscape, including chromosomes that Chris Roop and I discovered on our own that actually have the so-called site in both forwards and in reverse. Okay, so the biggest reason why this is probably not a fusion site has to do with the function of the gene that overlaps it. So there's a gene called the DDX11L2 gene. And you can do a search yourself. The evolutionists can do this themselves. And if you search the gene DDX11L2, notice here the website, go check out the gene, and you'll realize that it's highly functional. It's a long non-coding RNA gene that has almost 2,000 functional associations. If you go to this page, there's a bunch of drop-down boxes. You can click them. You can see what this gene is co-expressed with, what it's associated with. And there's a lot of function and activity with this gene. As a matter of fact, when it comes to long non-coding RNAs in general, there are many more. There's over twice as many long non-coding RNAs in the genome than protein coding genes. Remember, you have DNA transcribed into RNA, translated into protein. DNA flows into RNA, translates into protein. And so, yes, you have a lot of protein coding genes, but you also have a lot of regulation in the genome where you have DNA to RNA and you have a functional RNA product. So yes, it's never translated into protein, but it's translated into a functional RNA product. And these long non-coding RNAs are the key to understanding genome function. I like to look at it this way. Protein coding genes are basically the building blocks and are therefore more similar between creatures. If you were to compare all mammals, let's say all vertebrates, you're going to find a lot of similarity in the protein coding genes. The long non-coding RNAs that are actually produced by non-protein coding sections of the genome, on the other hand, are more organism specific, or in technical terms, they are taxonomically restricted. And these genes provide the metadata for the building blocks that are the protein coding genes to function. And this is where we find the most dissimilarities between creatures. So yeah, if you just look at the protein coding sections of genomes, you're going to find a lot of similarities. But as you bring in your non-protein coding regions, that's where creatures are less similar to each other. These long non-coding RNA genes, they're found in the nucleus. They're also found in the cytoplasm, and others are exported outside the cell to other cells in structures called exosomes. These non-coding RNAs, and I've got a few papers here from the conventional literature, they function in numerous ways. They're critical to the cell and healthy activities within the genome. This paper here talks about how they are a diverse class of RNAs that engage in numerous biological processes across every branch of life. The evolutionary community, they invoke what they believe are the best arguments for common descent, but by invoking them, it demonstrates that they are out of date on the literature. Notice this. Gene expression patterns have implicated these long non-coding RNAs in diverse biological processes, including cell cycle regulation, immune surveillance, and embryonic stem cell pluripotency. These are important DNA elements. There is a growing body of literature from yeast to mammals suggesting the non-coding RNAs have an important role in chromatin state formation. The DDX11L2 gene overlaps the so-called fusion site, and it's a long non-coding RNA gene. As a matter of fact, you'll be interested to know that long non-coding RNAs, they've been knocked out in mice. So you can take mice genomes, you can knock out certain genes and see what the effects are. And these knockout experiments turn out to be lethal. They kill the mouse. That means these genes are essential and that the RNA products produced by them are important. As a matter of fact, this DDX11L2 gene, the site that it overlaps, comprises multiple start points for transcription where RNA polymerase II binds. This is an amazing source of genomic activity. And the critics want to oftentimes, in their criticisms, they want to compare the DDX11L2 gene to protein coding genes. Again, that demonstrates their ignorance, because it's well known that non-protein coding regions, your regulatory regions like your long non-coding RNA genes, they're expressed at lower levels and yet are still functional when compared to your protein coding regions. And so what they're doing is comparing apples and oranges. Their arguments are very unsophisticated, they're unimpressive, they're not actually engaging our criticisms or our arguments here, unfortunately. The genome has been described as being analogous to an RNA machine. And so this site, being a hotbed of genomic activity, makes me ask the question, how do you get highly functional and essential genes like the DDX11L2 gene by slamming together two chromosomes? That's the question I want to. They want to say, oh, well, the gene moved over the fusion site. Okay, show us empirically how that's possible, when that's happened. Again, this is post hoc, ad hoc rationalization. And you can just look for the function yourself, because they'll oftentimes say, oh, Dr. Jeff Tompkins, you know, he's a creationist, he's biased. But as I've shown earlier in this slide, go look up the gene yourself. You can find that it has 2,000 functional associations. And this DDX11L2 gene, it produces long non-coding RNAs of various different types. It's alternatively spliced. It has dual promoters. A promoter is a genetic switch in the genome, and it turns out that the fusion site itself is a second promoter for that gene that overlaps it. And so it's a genetic switch. And Dr. Tompkins here, he points out that it's expressed in at least 255 different cell or tissue types. He says it's also expressed, co-expressed. What that means is it's turned on at the same time with a variety of other genes, and is connected to processes associated with cell signaling in the extracellular matrix and blood cell production. Well, this isn't surprising. Remember earlier in the paper here on long non-coding RNAs, they point out their function in cell cycle regulation, immune surveillance, so on and so forth. And so this is a highly functional gene. Dr. Tompkins points out, even more amazing is that the fusion site is itself functional and serves an important engineered purpose. So basically what we find with the telomeric repeats are designed elements within a highly functional gene. When it comes to the so-called cryptic centromere, those alphoid repeats, we'll get into that briefly too, those are also overlapped by a highly functional protein coding gene. So what we see that the evolutionists say are ancient remnants of past fusion are actually functional DNA elements inside essential human genes. And so Tompkins points out the site actually acts as a switch for controlling gene activity. See, when it comes to looking for function in non-protein coding regions, like the long non-coding RNA genes, you have to look to co-expression, you have to look to epigenetic related factors, and that's where you discover how significant the activity is, because we can look at what genes that this gene, the DDX11L2 gene, is linked to, and what other genes it is co-expressed with. And this gene is linked to the expression of many other important genes, many other functional genes. And the evolutionists don't do this. They want to compare protein coding genes to these non-coding RNA genes. That's apples and oranges. And again, it shows that they are out of date on the literature. So he points out a wealth of biochemical data showed that 12 different proteins called transcription factors regulate this segment of the gene. One of these is none other than RNA polymerase II, as I pointed out. And RNA polymerase II is basically the main enzyme that copies RNA molecules from DNA in a process called transcription. He points out further supporting this discovery is the fact that the actual process of transcription initiates. So this process of transcription, DNA to RNA to protein, the transcription part of it, is initiated within the region of the so-called fusion site. Tells us those DNA elements that are there for functional reasons. Technically, he points out, we would call the activity in the alleged fusion site a promoter region. Promoters are the main switches at the beginning of genes that turn them on, and are also where RNA polymerase starts to create an RNA. Dr. Tompkins also gets into the degenerate nature of the fusion. Fusion site should be a lot larger, a lot more sequences. It should be more pristine. It was not predicted that it would be degenerate like that. And so I have slide after slide showing why the so-called DDX11L2 gene is not just noise, it's not just spurious, as Dr. Ken Miller has tried to argue in these critics. The fusion site sequence binds to at least 12 different transcription factors. We pointed that out earlier. It's alternatively spliced, and it's a hotbed of genomic activity. The DDX11L2 long non-coding RNA gene containing the alleged fusion sequence encodes several transcript variants expressed in, notice this, at least 255 different cell and or tissue types. And so when these evolutionists argue that it's just spurious, they're demonstrating that they've not actually looked into this objectively. They're just repeating talking points. The fusion site itself appears to be the functional start site for the shorter transcript variants. And I've got a lot of data on this. I'm just going through this as quickly as I can, because you can check out my articles on it. You can check out my debates. I like the way he explains this here. In summary, chromosome fusions would not be expected to form complexly transcribed and regulated multi-exon, alternatively spliced functional genes. That's a mouthful right there. But again, it's basically asking the evolutionists to answer this question. How do you get highly functional genes by slamming together two chromosomes? And just saying, well, the gene moved over the so-called fusion site. That's a rescue device. That's science fiction right there. And the reason why it's unlikely to be a fusion is because these internal telomeric repeats, but then also your so-called centromeric DNA, the alpha repeats, they exist within essential genes. It should be chaos. We should have chaos going on in those regions, because a chromosome 2 fusion should result in chaos, not function and not evidence for design. In summary, the suggested fusion site turns out to not be a fusion site. It is a key genomic switch within a highly competent gene. The gene that crosses the alleged fusion location is a long non-coding RNA gene. The action of this gene has been tracked using public databases. You can do this yourself, as I showed in the presentation, to numerous significant human tissues. The gene is associated or connected with several important networks in other crucial genes in the human body. And the internal telomeric sequences represent functional units important to essential genes. The last thing I want to touch on when it comes to the fusion is this idea of a so-called cryptic centromere. So basically, if two chromosomes fuse together, we should have two centromeres. Centromeres are essentially attachment sites in the genome where the cell division apparatus can latch onto the chromosome after it's been replicated. And centromeres are a very critical aspect of the genome. Having two centromeres would create an especially unbalanced situation. Therefore, if a fusion did happen in the distant past, we should find a disabled or a silent centromere in the chromosome 2 area. The evolutionary community have claimed to have found a hidden centromere in the so-called fusion site. But what scientists actually discovered was an exceedingly small sequence when equated to real functional centromeres that exists in about 250,000 bases. This so-called mysterious centromeric sequence was only about 41,000 bases long. So much like the telomeric DNA, it's a lot smaller or degenerate than it should be if it really is an example of an ancient fusion. The sequence also had interruptions in it that were not centromeric-like. And so this unsubstantiated fossil centromere is located, as I pointed out earlier, in a large functional protein coding gene. So it's very difficult to imagine that this fossil centromeric DNA or signature exists inside a functional protein coding gene. It's also evident that the sequence claimed to be a cryptic centromere and evidence for a fusion event in the past is, as a matter of fact, a functional component of a well-designed gene. And so your two key elements, your telomeric DNA and your so-called centromeric DNA, they are located inside highly functional genes. And parts of the implied centromeric DNA actually codes for an aspect of the protein itself. And so since this site is claimed to be the leftover remnants of a historical fusion, since they are engaged in important operations within the cell, it's highly improbable that they could have been derived through a mysterious genetic blunder, because that's exactly what a fusion is. It's a blunder. It's a genetic mistake. These elements looked at by evolutionary scientists and proponents of common descent as evidence for human evolution turn out to be functional features inside human genes. Okay, I'm going to quickly go over endogenous retroviruses, and then we're going to wrap it up. I've done a whole book on this. I've engaged in multiple debates on this, including debates with people who've read my book, spent a couple months prepping, and then I engaged their criticisms. You can find on my channel a series of lectures I've given on endogenous retroviruses, hours and hours long. And so I'm just going to cover this really briefly. What are endogenous retroviruses? According to the standard evolutionary story of endogenous retroviruses, an ERV is a stretch of DNA found in your DNA that got there when one of your ancestors was infected by a retrovirus. I've got all the sources documented, demonstrating this is exactly what they believe. You can see the source here. They also believe that ERV elements are the ancient remnants of past viral infections. Some evolutionists will refer to ERV sequences as fossil viruses. Evolutionists acknowledge that for the most part of these ERV sequences, the infections would have occurred millions of years ago. So basically, they have exogenous retroviruses like HIV. It's called exogenous with an X because they're occurring from outside. And so they'll say that a common ancestor between humans and chimps would have been infected with an exogenous retrovirus. This retrovirus would have integrated into the germ cells, sperm or egg, reproductive cells, which means they're passed on. Once they're passed on, the progeny's DNA will have this viral DNA in every single one of its cells. And then you'd have a split, humans and chimpanzees. That common ancestor would split into two lineages, and they would take those viral sequences with them. And so evolutionists compare those sequences that are in similar genetic loci or locations, and they say they must have been inherited from past common ancestors. The question is, and the question I like to ask is, are these really the ancient remnants of past viral infections? Or do they represent functional units of DNA? Well, firstly, a retroelement is a sequence that moves around the genome by reverse transcription. But we have many retroelements, as you can see here in the visual. Endogenous retroviruses are just one of those retroelements, and they are tightly integrated in the genome. There's evidence that ERVs currently actually can't really move around. They're one of the non-mobile retroelements. But the ERV does have the classic retrovirus design, as I'll cover in just a second. 42% of the human genome actually comprises these retroelements, and they're broken into different classes. One of these classes or types are endogenous retroviruses. And so what do we see with an ERV? Well, with an ERV, we have host DNA duplicated on either side. Okay. Actually, here's different types. You got signs, lines, retrotransposons, and then your endogenous retrovirus at the bottom. And so I want you to just focus on the structure of the ERV. So we basically have the host DNA duplicated on either side of the provirus. We have two LTRs. Those stand for long terminal repeats. Then we have a gag, a pole, and an envelope protein. Stands for ENV. That's the structure of the ERV, which is the same structure to retroviruses. One question I like to ask the evolutionists, and they never have an answer, is what came first, the retrovirus or the host? Because retroviruses actually require a host to replicate. The creationists would argue that retroviruses originally came from the host because of the fall. And even the secular literature has a model called the escapee hypothesis. And so we would argue that bad retroviruses escaped from host genomes, but those ERV sequences, for the most part, are created units of DNA function. And so the critics will ask, well, why do they share sequence similarity to retroviruses? Well, it's funny because a couple of the functional roles associated with ERVs has to do with the immune system. And so here's a recent technical paper. And in my book, I document dozens of points in the secular literature going over the functional roles with ERVs. And so one of their functions is to protect us from viral infections. And so they actually act as antiviral protectors within the genome. They're like antiviral programs that God has front-loaded into the genomes of biological organisms. And so if bad, harmful viruses enter the genome, we have these ERV units to basically fight off your incoming viruses. But a way they do this has to do with their sequence similarity to the retroviruses, because they're involved in what's called viral mimicry. And one of their roles is tumor suppression. So basically, transcribed ERV sequences, they give off the impression that cells are being invaded by viruses. And as a result, this allows the immune system to target more damage. This is actually mediated by what's called the superhero protein of the genome, the P53 protein. And so the immune system targets these tumor cells with more destruction and thereby killing the tumor cells because the ERVs are performing viral mimicry. Well, in order for them to perform that role, they have to look like bad viruses. It would be like an undercover FBI agent infiltrating the mob. Well, is he going to show up with his FBI badge and his FBI uniform? No, he needs to play the part. He needs to infiltrate and play the part. He needs to look like somebody who would belong to that organization. And so a lot of these ERVs, yeah, they do have sequence similarity to retroviruses, but it is necessary for many of their functional roles. And so the answer to the question, do ERVs represent functional units of DNA or the ancient remnants of past viral infections? Since they're involved in determining cell types, they're also involved in embryological development. They act as antiviral protectors. They function in gene regulation, gene expression, and they perform viral mimicry. So the answer is they're functional units of DNA. And so why do we find these ERV sequences shared between humans and chimpanzees? Well, for the same reason why we find engines shared between all modes of transportation. This is good design. Why do we share more with the chimpanzee than we do with the dog? Well, overall, just stand back. We share more in terms of morphology and anatomy with chimpanzees than we do with dogs anyways. So that's not a big surprise. And so here's just a few papers documenting what I'm saying, and then we're going to wrap it up here. So HERVs, if you see an H there, that stands for human-specific ERVs. They appear to play important roles in physiology, fetal development, and human evolution. Notice this. I find this one to be hilarious. They say if the accidental infection, so they're assuming that the infection took place, we'd argue no, this ERV was created the way it was. So they say if the accidental infection of a mammalian ancestor by an exogenous retrovirus had never occurred, the placenta and the mammals that produce it, including humans, would have never existed. I like to exist. I hope the listener likes to exist. My awesome host, Brian, likes to exist. What are they saying here? These endogenous retroviruses are necessary to exist. So is there any surprise that all forms of life share these DNA units in common? They point out they act as mediators of normal biological processes such as cell differentiation, regulation of gene expression. And I could spend the next 2 hours going over all the many papers that talk about how they regulate human transcription on a large scale. Again, they act in the immune system. Notice this paper. Scientists identify a new beneficial function of endogenous retroviruses in immune response. Transposable elements have had a substantial and sometimes beneficial impact on host genomes in several ways. So another important function of just transposable elements in general is they can move around the genome from one chromosomal location to another in introducing novel genetic variation, or even just turning genes on and off. This paper here talks about how they are a prolific source of tightly regulated biochemically active non-coding elements. Curves appear to play important roles in physiology, fetal development, and human evolution. That is assumption-based because they think the variation that can be induced or created through these ERV elements will help with forward evolution. But no, all we get is variation within the same kinds of creatures. And so as you can see, paper after paper, here's what I was talking about earlier. Endogenous retroviruses are emerging as promising and therapeutic targets in cancer because of their viral mimicry function. Notice this paper. Endogenous retroviruses act as an unexpected ally in the regulation of human immune systems. You wouldn't want to live in a world without endogenous retroviruses. So here's my last slide. As a response, the evolutionists will argue, well, these functions were co-opted. But that is an assertion, that is a claim, that is their evolutionary philosophy, because we've never seen an exogenous retrovirus integrate into a host genome, integrate in the right way, which would be in the reproductive cells, to be passed on. Then mutations have to destroy the negative aspects of your viral DNA. And then through mutations or co-option, a novel function needs to come about. That's never been observed. We've never seen a non-functional endogenous retrovirus go from non-functional to something extremely functional in embryological development. And so when these evolutionists assert co-option to explain these essential roles associated with ERVs, you know what you say? You give them the thumbs up and you say, cool story, bro. And so that wraps up my presentation. I know there was a lot there, but there's still so much more, especially when it comes to ERVs. And that's why I highly recommend for people to check out my book, check out my debates. I've done an entire lecture series where I also go over testable predictions that I've made, novel testable predictions on ERVs, specifically the nature of fixed ERVs versus unfixed ERVs. And so with that, Brian, I really appreciate your patience, brother. And I guess I'll stop share and hand it over to you.
Awesome. All right. That was very, very comprehensive. You actually answered most of the questions I had. So that was really
Feel
good.
free to re-ask him, brother.
All right. So that. On the ERVs, are you saying the ERVs actually have an actual function within us, that they're not like this useless thing that was passed over due to common descent that they believe in? Is that correct?
That's correct. And many of their roles, brother, they're essential. As in, without endogenous retroviruses, as you can see from that paper, they talk about how mammals wouldn't exist, because endogenous retroviruses are actually critical to pregnancy and embryological development. And so without ERVs, there would be no embryological development. I'll say this. In the mouse genome, they did knockout experiments where there's a specific class of retrotransposon in the mouse genome. They take it, they snip it out, they get rid of it. Well, the mouse is developing. Once they knock out that retrotransposon, you know what happens? The mouse dies, because the mouse and its development is based on that retrotransposon. Without it, it won't exist. And so they're needed. They're necessary for all of biological life.
Well, let's start by talking about the DDX11L2 gene.
Sure.
A lot of evolutionists claims that that is a pseudogene. Well, first of all, okay, what is junk DNA? What is pseudogene? And is this a pseudogene? Is this a, how they define a pseudogene?
Good question. And so the DDX11L2 gene, I went over several slides, as you know, in the presentation showing why it shouldn't be categorized as a pseudogene. So a pseudogene is basically a broken version of a protein coding gene. So you have a protein coding gene that was hit with mutations, or it was subject to retrotransposition. There's different kinds of pseudogenes, unprocessed pseudogenes, processed pseudogenes. And so they'll say that this is a gene that's broken. It can no longer produce a protein. When I debated Dan Stern Cardinal on this issue, he asked the question, well, does this produce a protein? And I said, well, that's based on your evolutionary assumption that if it doesn't, it's non-functional. Because as I pointed out, these long non-coding RNA genes, which the DDX11L2 is, they're critical. They're key to understanding function in the genome, and they're functional in just a huge number of ways. As a matter of fact, they've knocked out these long non-coding genes in mice, and they've turned out to be lethal. That tells us we need them. And so your protein coding genes that are hit with mutations and become what they call a pseudogene, they would assume that these pseudogenes are non-functional. They're broken genes, basically. But we find almost all types of pseudogenes to have necessary functions. A lot of these so-called pseudogenes actually help to regulate the function of their protein coding counterparts. And so there's actually a lot of network connections going on within these genes. And so, no, I wouldn't call it a pseudogene. It's a functional DNA element, and it has a lot of activity. It has close to 2,000 functional associations. And so they're the ones that are assuming that this was once a protein coding gene, and now it's been basically hit with a series of mutations, or however they explain it. And so now it's a non-functional gene. And so they'll say that the activity associated with it, they'll say that it's spurious or it's noise. It's not really doing anything. But we know it's expressed in at least 255 different cell or tissue types. It's also co-expressed with a variety of other essential genes, and it's connected to these processes associated with cell signaling. And so it's just a weak argument to say that it is a pseudogene or the function is just spurious. I think that's been debunked. Good question, Brian.
Did they is a pseudogene like junk DNA, where they consider it like a leftover from our evolutionary development and it's now a useless gene? Is that how they look at it?
Exactly. So the evolutionary model, they would look to, as you know, descent with modification. And so if we've been evolving for millions of years from past common ancestors, we share, an ancestor with fish, according to them. Somebody like P.Z. Myers would say we're still fish. Well, then they would expect to find many shared genetic mistakes over time, where common ancestors have been hit with mutations that have broken down protein coding genes, and those mistakes have been passed on. So when they look to pseudogenes in the same genetic locations, Brian, between humans and chimpanzees, they argue that those are there because those mistakes were inherited from a common ancestor in the past. But we point to the scientific literature, and I've got dozens of papers that actually show no, these are not genetic mistakes. They are actually necessary to sustain healthy cell processes. And it's akin to this. If I were hired to be a proofreader in Russia, let's say, so I got to read a book and I got to point out what's a mistake and what's not. I don't speak Russian, so I'm not going to be very good. And that's basically what's going on in the DNA language, is these evolutionary scientists, they're pointing to these sections in the DNA and they're saying, oh, this is broken. Oh, this isn't functional. Oh, this is a viral fossil. We're actually finding out in all of those cases, these are actually functional DNA elements, whether it's an ERV, whether it's the DDX11L2 gene, whether it's gene deserts that they'll argue are junk DNA. I pointed out in my presentation the ENCODE project. They discovered that over 80% of the genome is active. It has evidence for biochemical activity. We would say most of that activity is there for a reason, because if it wasn't, that would be wasteful of energy and resources for the cell. And so natural selection should have got rid of that genetic baggage over time, or there should be processes in the genome that suppress this activity if it's really just noise. And so their response is weak. The evidence suggests that the activity is there for a reason. And that overturns the junk DNA paradigm.
Just for a point of reference, the fusion site, if a fusion did occur, it cannot be in an active gene. That's the idea, that it cannot be in an active gene, because if it's in an active gene, then the fusion could not possibly have occurred. Is that right?
Yeah, a couple things. You don't get highly functional genes by slamming together two chromosomes. So if you had two ape-like chromosomes that came together and fused, basically, you're not going to get a highly functional gene that way. But what we should expect within the region, we should expect chaos. But what do we find? We find functional units of DNA. Those telomeric repeats are associated with gene expression. We also find that the so-called centromeric DNA, which in technical terms is called alphoid repeats, that is overlapped by a protein coding gene that Dr. Jeff Tompkins has pointed out. And so it's hard to believe that these DNA elements are situated within a region of the genome that experienced a genetic blunder like a fusion. But what they'll do, Brian, is that, like Erika, she argues, well, if that DDX11L2 gene is functional, it could have moved over at some point on top of the fusion site. But my question is, where's the evidence for that? Show me something similar where a highly functional gene moved over this region, and now that region, because remember, the region acts as a second genetic switch or a promoter for that gene. And so there is a network that's connected there. You know, the genome, it's four-dimensional, as Dr. Rob Carter points out, it's four-dimensional. There's multiple overlapping codes. It's full of meta information. It's nested. And so you have all these elegant networks within the genome that are connected, that if they were not connected, they were broken through mutations, they wouldn't function. And so this is all connected, which means no, the gene didn't move over the site. The site itself is actually helping with the function of the gene. Does that make sense, Brian?
Yeah. Telomere fusions, they normally don't happen in nature unless dealing with a cancer or something like that where cells are going crazy. But telomere fusions don't normally happen. Is that correct?
It's very rare. Usually it's not telomere to telomere fusion. Usually fusions
Centromere or telomere or something like that.
Yeah. Usually the fusion would have satellite DNA associated with it. We don't find that in the human chromosome 2 area. Now, although it is rare, barely ever happens in nature. Apparently, there's been two purported examples. One in pigs and another in equids. Both are inferred. If you read those papers, there's not a lot of detail in terms of the genetic content of those regions. And they say, well, most likely this occurred due to a telomere to telomere fusion, but this wasn't observed in nature. They're seeing the signatures in the genome and they're inferring that it occurred in the past, and they're inferring that it's a telomere to telomere fusion. And so for sake of argument, if it is, it's still very rare. The fact that they can only find two examples in all of the documented fusions tell us that if this really is a fusion in the human line, it would be the type of fusion that is extremely rare.
Actually, in a little bit, we're going to be looking at a couple clips from Erika's response to you on that. One of them will have that. I guess what I'm calling her pig argument.
Yeah.
I personally found the argument kind of circular, but we'll get
Yeah.
into that, I guess. But any given case, the fusion site, the size of the fusion site is allegedly, or it is, seven, or what they claim on the fusion site, 798 base pairs. Now, telomeres, and you pointed this out in your presentation as well. Telomere is typically what, 10,000 or 5,000 to 15,000, something like that, of
That's
base
right.
pairs. And so you would expect this fusion to be really, really big if it happened. I believe I read, you might have said it in your presentation too, but I know I read it in some of the stuff that you sent me, that that one of the claims that evolutionists make is that the fusion size lost some of that information or something like that. Is that correct?
The fusion, the 798 base pairs, the evolutionists will argue that the reason why it's so small, it's so degenerate, even the evolutionary community, I showed one citation where they asked the question, why are the arrays at the site so degenerate? There's a couple answers that they give. Since telomeres act as end caps of chromosomes, they're there to protect from fusion. And so they would argue that those telomeres should have degraded before the fusion happened. And so when the fusion happened, there wouldn't be all that much telomeric DNA there. Or they would say if there was a fusion, but there was a lot of telomeric DNA there, that it would have been lost over time. Since we may be looking at 4 million years since the fusion, within that 4 million years, they could argue that the telomeric DNA, a lot of it's been lost or hit with mutations. And so it's degraded from a more pristine point, they'll argue.
What do they mean by degradation exactly? Because Erika kept saying that in her response video that it has to be degraded. And you seem to suggest that this is a post hoc ad hoc claim because originally they said the opposite. Is that correct? Am I understanding that correctly?
Right. Right. They never predicted it. That's why the question the evolutionary authors ask is, why is the area so degenerate? So if what Erika is saying is true, that this should be expected, why didn't they ask the opposite question? Or why didn't they just assert, well, the area is degenerate and this is what we expected? But even if you want to give them the degeneration, healthy telomeres are between 5,000 and 15,000 bases long. And so if there is no degeneration, you're looking at somewhere between 10,000 and 30,000 bases for a signature. Let's say it degenerated quite a bit. Well, we should still see something in the thousands. Why is it less than 1,000? Why is it only 798 base pairs? Why are those internal telomeric sequences directly associated with the hallmarks of gene expression? How is that possible? We should see chaos. And so even if you wanted to give them the degeneration argument, I would still argue that we should still find more than the 798 bases. But again, I would just point them to their own literature where they ask the question in surprise, why is the area so degenerate? I'm really curious why they didn't just say, oh, it's degenerate. And that's what we expected.
Would you say their claim of degenerate is really assumed in terms of, because they assumed that's the
Right.
size of the fusion area, so they assume it was degenerate.
And you asked, what do they mean by degeneration? And so basically, it could have been more pristine. Maybe there were more sequences when it happened, but over time, it's been hit with mutations and it's basically garbled up the signature. It's ruined it. It's destroyed it. It's degraded it over time. And so maybe according to them, a couple million years ago, there were more head-to-head arrays, TTAGGG, 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 but maybe over time mutations have hit certain spots within those tandem repeats, and so it's disconnected them. It's made them less pristine, is what they could argue. But yes, like you said, I think it's circular. We've all got presuppositions, and they are assuming that there was a fusion. They require a fusion to explain the discrepancy in chromosome numbers. Humans have 46, the great apes have 48. And so they require this. Ken Miller said if there was no fusion, there was no human evolution. So they're pretty desperate in this argument. They really need it. And so though it's degenerate, so they'll say, well, it had to be degenerate, even though they acted surprised. And so, yes, I think it's all based on evolutionary assumptions. It's all based on the assumption that this is a fusion. Now we have to explain all the data that contradicts the idea that it's a fusion.
One of the arguments that Erika did, and including her response video, she accused you of a straw man argument. She said that because the other half of her argument was in high density. Now, interesting enough, though, she was responding to a particular video that you had made, responding to her, and and when I was reading some of the stuff that you put out there, including you have a response, particularly on, I think it's on your website. I think that's where it probably originally was. You might, I think I might have got this from your website, so I don't think this is one of the things you sent me. But you actually do talk about the high density. You actually do talk about the claim of the high density and so forth. But first of all, what do they mean when they say that this is in high density and they claim that it's not in high density in other places? And two, is that true?
Yes, good question. Before I answer that, I wanted to, so this book right here, Contested Bones, if you can see it in front of me, it is Christopher Roop wrote it, him and Dr. John Sanford. Chris Roop is the brother in the Lord that I discovered the chromosome 9 with. And so on page 326, they make a good point about the degradation. So they say, it might be argued that since healthy telomeres help prevent end-to-end fusions, maybe both of the two fusing chromosome tips were severely degraded, right? Like the evolutionists say, with both telomeres eroded to near extinction. This is feasible, but it is a very weak argument. It seems unlikely that both of the fusing chromosomes were degraded to exactly the same point, yielding a minimized and semi-symmetrical fusion site. The reduced site of chromosome 2 compared to the two putative fusing chromosomes also strongly suggests that if there was a fusion, it involved deletion of both chromosome ends as observed in illegitimate crossing over events. So a couple of points there. They point out what I pointed out, that even if there was a fusion, the separate ancestry model could explain it best. But secondly, their argument about the degradation is pretty weak, because why would both chromosomes be degraded to exactly the same point to create a semi-symmetrical signature, basically? So there's a lot of post hoc, ad hoc rationalization going on. And I think Chris Root makes a good point there in his book. So your next question about the density, was it basically, why is she arguing that it's more dense in chromosome 2 versus other spots?
What does she mean by it when she says that it's in high density, and two, whether it's actually found that way or needs to be found that way in order for your arguments to pan out?
Great question. I think it might be good for your audience to have a visual on this one. So I can pull up my article with Chris Roop if you'd like.
Go ahead.
Okay. So I click share screen and let me.
I see it.
Okay. Trying to get this out of the way to get to my. Is there a way to move this? Pause, share, remote control. Okay.
Oh.
It looks like it went away. Maybe you did that. I'm not sure. But okay. So here's the article I wrote based on our findings. And so what she means by density is, here's the fusion site. So you got 798 bases, right? TTAGGG, that's your telomeric DNA and forward. The reverse complement, because DNA is double-stranded, is CCC-TAA. So we find at the fusion site, we have one example of TTA-GGG here. Then we have a lot of degradation, where we don't get another example of TTAGGG until many DNA letters later. So we got 1, 2, 3, we got about 9 here. But you'll also notice that the majority of them are not connected. They're not head to head. There's one head to head here, but most of them are isolated. Same with the CCCTAA. But what she means by density, Brian, is notice the size of this. So it's very dense. There's a lot of these TGAGGG. There's a lot of the telomeric sequences as compared to anywhere else. So if you look at the spot we found, this is in chromosome 9, Chris Roop and I, you have TAGGG, TTAGGG, TTAGGG, TTAGGG. So notice how pristine this is. They're all head to head many times. They're connected. Over here at the so-called fusion site, they're found in isolation. They're very disconnected. It's not pristine. And then we get the head-to-head organization here, where you get the reverse complement, CCC-TAA, CCC-TAA. But this is not, I think it's about anywhere between 40 and 45 bases. So it's not dense compared to your so-called chromosome 2 area, where you have 798 bases of this type of DNA. There's not as many here in the chromosome 9. And then we find them in many other spots, but we don't really find too many examples where they're head-to-head, TTAGGG and then CCC-TAA. We do find them, but usually just an instance here, an instance there. We do find the internal telomeric repeats like TTAGGG or CCC-TAA in multiple places within the genome, as I showed in my presentation. But the chromosome 2 area has the highest density, or the largest number of them. Does that make sense, Brian? Does that help answer your question?
Yeah, the first one. With the second one, I want to know whether or not if it requires. She thinks that the high density proves that it's a fusion site.
Right.
What would you say to that?
Well, I would argue, firstly, I would agree that it is the highest density of these sequences in the genome. Okay, so we find, I really want to give your audience a visual. So I covered this in my presentation. Notice this. Here are hits all throughout the genome on your internal telomeric repeats, your TTAGGG and CCC-TAA. So you can even see that your chromosome 2 spot, notice how many are situated here in the middle. So we find them everywhere. That's not to say we find them in a head-to-head organization everywhere. That we find in chromosome 9, like I showed. And then we have single examples in multiple other chromosomes that I document in the article there. But your chromosome 2 does have the most of them. It is a massive chromosome. It's the second largest chromosome in the genome. Notice 21, 22, 16. They have a lot of these telomeric repeats, and they're small chromosomes. The chimpanzee genome barely has any. Notice how absent these are from the chimpanzee genome. And so this tells me that these units, these internal DNA units, are there for functional reasons. And since they are directly associated with hallmark gene expression, hallmarks of gene expression, as in the type of transcription factor binding and gene activity happening at the so-called the purported fusion site, this is occurring genome-wide at several of these interstitial telomeric repeat sites. And so this function might be associated with the nature of humans and how we differ from chimpanzees. We understand we differ, especially intelligence. We differ in so many ways to the chimpanzee chromosome. And so it might be this way for human-specific reasons, since they seem to be human-specific DNA elements. But anyways, yes, the chromosome 2, it has the highest density of these repeats. They want to assume it's because of a fusion. But I would argue that this is a logical fallacy, and it is, first, it's circular, because Erika is asking for us to find this exact dense region, the 798 base pairs, elsewhere in the genome. Well, why would we find that exact same unique signature elsewhere in the genome? She wants us to, so she's assuming it's a fusion site and saying, show me that exact same fusion site somewhere else in the genome. Well, we can show you very similar signatures, but they're not going to be as dense. And so she's going to point back to the density. But much like how the critics, Brian, they'll argue, well, the universe looks old. And we respond with, well, wait a minute, we only have one universe. It's not like we have another universe that looks young. And so when we compare it to this universe, oh, it looks old. No, we only have one universe. And we have the data that exists in this universe to analyze and examine. Well, we only have one Earth. We only have one human genome. We don't have Earth 2 and Earth 3 with other human genomes that we can look at and compare to this human genome. And so, yes, it's dense, but it could be dense for other reasons than a historical fusion event. If I go outside and the grass is wet, is it wet because it rained? Well, it could be wet for other reasons too. It could be wet because somebody had the sprinklers going. You know, it doesn't have to be wet because it rained. There's multiple reasons for that.
Or dew in the morning.
Yeah, right. Dew in the morning. You know, there's many reasons. I would argue that because this area was a surprise to the authors that asked the question, why is the area so degenerate? Which I'll just pull up for the audience sake here. They asked right here. This is not a young Earth creationist, right? This is a prominent paper examining the chromosome 2. And they're asking if the fusion occurred within the telomeric repeat arrays less than 6 million years ago, why are the arrays at the fusion site so degenerate? Again, they didn't say, oh, it's degenerate and that's what we expected. No. And they go on to give what they believe are possible reasons why it is degenerate, but that shows that they weren't expecting this area to be degenerate. And so, yes, it's dense, but it's circular to assume it's dense because of a historical fusion. Because now, if you want to talk about a fusion, it's actually not dense. It's not dense if we're going to compare it to what a fusion should look like. But it is dense when we compare other areas in the genome with these DNA elements. And so I would argue it's very arbitrary. It's very subjective for Erika to say the reason why it's dense is because it is an example of a historical genetic blunder. Does that make sense,
Yes,
Brian?
yes.
And what do you think about that?
In a couple or in a few minutes, we're going to deal with a couple clips from that response video that she made, and basically she's going to cover something in a paper on pig evolution. So I want to be able to discuss that with you, and it's probably going to deal with some of that as far as what she claims, because she's claiming that the same evidence can be found in other areas that they think is fusion, and therefore that must make that true. Right. Well, if you want to go ahead, you can go ahead and respond to that. Does that, if they could find that type of like in high density, in telomeric repeats, that sort of thing, if they find it like that in other areas they think is fusion, does that automatically mean that this must be evidence for fusion?
That's an excellent question. I can respond to how you reiterated her arguments, or did you want to play the clips and I'll respond directly to what she's saying?
I'll play the clips in a second. I just have another question before we get into that on centromeres. One of the arguments she makes is that that they have found like 2 centromeres, because each a chromosome normally just has 1 centromere, and so they claim that there's a second centromere. So that must mean that there are 2 different chromosomes fused together. Now, you did mention that in your presentation on the centromere. Is it my understanding that you're claiming that the evidence that they're claiming for a second centromere is weak?
Yes, good question. I shared my screen again, so let me know if it's coming through.
Yes, it
I've
is.
got a whole cool. I've got a whole section on the so-called second centromere. And if I go down to it, okay. So this so-called area that they think is a deactivated centromere, it's actually situated inside an actively expressed protein coding gene. Dr. Tompkins addresses it here. And so it's very difficult to believe that this is the ancient remnants of a deactivated second centromere if we have functional units that exist within a functional gene, because it actually turns out that some of this so-called fossil centromeric DNA, it functions with the functional protein coding gene that overlaps it. And right here, he points out that Tompkins says another serious problem with the alleged cryptic centromere is that its human alphoid repeat DNA sequence does not closely match homologous chimpanzee centromeres and chromosomes. And he points to a paper here where he says, in fact, the differences in human versus chimpanzee alphoid repeat arrays are so profound that Half and Willard, in a later publication, stated, so this is from the conventional literature, they say this implies that the human chimpanzee sequence divergence has not arisen from a common ancestral, and then it goes through the satellite repeats. Of particular interest to the idea of a cryptic centromere is the fact that human chromosome 2 had 6 internal regions bearing alphoid repeats outside the centromere. Clearly, the alphoid region at the alleged cryptic centromere site is not a unique or an uncommon feature in the human genome outside centromeres, especially in chromosome 2. 13 other human chromosomes also showed multiple regions of alphoid repeats outside the centromere. And you can see, similar to the telomeric DNA, Brian, you can do the same thing with the centromeric DNA. And so you can find all of the hits in the chromosome 2, but then you also find them elsewhere in varying degrees and varying amounts. But you do find, chromosome 4 here. I assume your critics like Erika are going to make the same argument with chromosome 2, that there's the most found at this area. But again, I would go back to the fact that your so-called telomeric DNA is situated within a functional long non-coding RNA gene. Your centromeric DNA, so-called, is situated inside a functional protein coding gene. And both portions of functional DNA are directly connected to those genes, as in those genes depend on those functional units. And so I would argue, I would ask the question, how come it's not chaotic? Why isn't it a mess? Because if this really was the ancient remnants of a fusion, we should see chaos going on in these regions, not function, not organization, and certainly not significant genomic activity. Does that help, Brian?
Yeah, you would say the second centromere is therefore not really a centromere at all. Is that correct?
Right. I would argue that the so-called centromeric DNA at the site is actually just another example of functional DNA units necessary for the function of the gene that overlaps it.
Because
Functional features inside human genes, basically.
if it was what they were thinking, it would be a silent centromere and have no function at all.
Right. I wouldn't expect there to be function. I would expect chaos. I would expect disorganization. I would not expect to find such elegance. We see an elegant system. We have DNA elements, and then we have genes that overlap these DNA elements, and they're both connected and necessary for the overall function of the human genome. I wouldn't expect to find that if this was all the result of a genetic blunder millions of years ago.
All right. I'm gonna go ahead and show two clips. Um, Let me go ahead and share my screen. Oops. I need to switch it back. Give me a second. All right. So we're gonna play a few clips from Erika. All right, let's play it. Are you ready?
Yes. Yeah.
The density of the telomere-telomere calling cards in the center of human chromosome 2 is half the argument, which is something that I've mentioned in every video that I've made on this subject. You're probably going to find isolated telomeric repeats once or twice elsewhere in the genome. The telomere calling card is 6 base pairs long, and the human genome is several billion with a B base pairs long. You're probably going to get the telomere calling card by random chance a few times, completely excluding duplications and inversions and other genetic shenanigans. But things actually get worse because remember.
All right. Basically, it's the same argument about density. She's accusing you of straw man argument. You're going to get this a couple times. The object of it is that yes, you're going to find it somewhat, but that still doesn't disprove the fusion thing, which is kind of strange because originally she said that if you could find that, that would disprove it.
Yeah. Well, she's never going to admit this, but her quote unquote challenge was issued in a very sloppy manner. And so what I mean by that is there were times where she would argue that you won't find the telomeric sequences in forward and reverse, TTA, GGG, CCC, TAA, in mirror form and in high density. But then there's other times where she just says, and I document this in my video response to her, where I respond to her original video, and quote, she says almost exactly, you will not find these telomeric sequences in forward and reverse in mirror form. And that's it. She didn't say anything about the density. And so her challenge was kind of sloppy, but I find it funny now that she is doubling down on the density portion being part of the challenge, because to me, that demonstrates how circular her thinking is. Because if part of her challenge is to have us find, so she basically wants us to take the fusion site, right? The 798 base pair region, and she wants us to query that in the human genome and find somewhere else where we find that exact sequence. That is assuming that region in the chromosome 2 is a fusion in the first place. And that is assuming that this should be a common occurrence, that we find this exact signature, those 798 base pairs, in other places in the genome. So we found places in the genome, one specific place, the chromosome 9, where we have many instances of TTA-GGG, 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 and it connects with the CCC-TAA. And this is pristine. This is more pristine than the so-called fusion site. And so, but then she wants to say, well, it's not as dense. Okay, well, then we can talk about the density and why it's arbitrary and subjective. But if the density is part of the argument, she's only proving how circular her reasoning is, how circular her argument is what I would say. So I don't think she'll ever admit to that. But when I respond to her, I play her word for word, only saying that we don't find anywhere in the genome where the TTA-GGG and the CCC-TAA are connected.
If density was part of the argument, then she should have added that to the statement.
Right. If the density is 100% necessarily part of the argument, then she always has to point that out. Don't forget, because apparently that's a major part of the challenge, according to her. But there's been at least that one instance where she did not say that the density was part of the argument. But to make the density part of the argument demonstrates how strongly the evolutionary paradigm has infiltrated her thinking, basically.
And would you also say that that her claim that, oh well, maybe you could find it somewhere, is just a way to justify the fact that this is not a unique occurrence?
Well, it tells me that she didn't really do the greatest job in terms of her research when she issued this challenge, because why didn't she look for other instances in the genome where we have the TTA, GGG, CCC, TAA? She wasn't aware of the instances that we documented, including the chromosome 9, with a fairly pristine example of what we see in the fusion area in the chromosome 2. What she's saying now, she never said before. She never said, well, just by chance, we'll probably find your TTA, GGG, and your CCC, TAA in a head-to-head organizational pattern.
Yep.
She never said that, but now she's saying it. So to me, she's doubling down, I feel.
Also in this video, she said that the part of her statement is that if this is a unique occurrence, or is this a unique occurrence, or is it occurred somewhere else? But now it's like she's saying, okay, maybe you'll find it somewhere else, but that don't mean anything. When in reality, that's a big deal if you find it elsewhere.
Well, I showed earlier from my article the nature of your chromosome 9, where you had several instances of your TTA, GGG in tandem repeat. TTA, GGG over and over again, and then connects to the CCC, TAA. And then she argues, oh, but this is too pristine to be a fusion. So now she's saying it looks too good. It needs to be degenerate, like we see in the chromosome 2, even though her own side in the papers themselves never predicted it to be degenerate. They actually asked the question, why is it so degenerate? As in they were surprised. Why can't the chromosome 9 area just be a fusion that did not experience as much degeneration? I just see a lot of circular reasoning coming from Erika and the evolutionists. That's not to say that I am arguing that the chromosome 9 is a fusion. I am just saying we find these DNA elements. We find them in varying amounts, in varying degrees, in varying densities all throughout the genome. They're associated with function. And the chromosome 2 area, we find the highest density of them. And so the debate comes down to, why do we find the highest density of these functional DNA units at the chromosome 2? Right. And then we can discuss that for a couple hours,
You would say that this is evidence that it's not, that this does not mean that it's a fusion site, because that this evidence is there. It would not require that, because there are other places that nobody thinks is a fusion site where the same thing appears.
Exactly. Why aren't they arguing that chromosome 9 is an example of an ancient fusion? Because we find the same head-to-head organization. We find it in mirror form, TTA, GGG, CCC, TAA, just like we find at the chromosome 2 spot. But the chromosome 2 spot is in higher density, but compared to what? Because if we're going to compare it to what a fusion should look like, then it's not dense at all. It should be even more. It should be in an even higher density. But this is also why I like to point out, Brian, that even if there was a fusion event, remember, this would have been a near extinction event. That one person who experienced this fusion would be the ancestor of all humans today, you, me, Erika, which means all other lineages, all other people groups on the globe at that time, they would have had to been wiped out for this to become fixed. And so if there was a fusion, what model best explains it? Ours, because we have the flood bottleneck and we also have the Babel bottleneck where this can become fixed. Because, I will say that the whole density argument is an interesting argument. And I think as creationists, we would do good to continue researching why that area is so degenerate. That's why I like to point out, even if there was a fusion, it's not discriminatory evidence for human evolution. But because the evolutionists are doing such a bad job in addressing the functional nature of the DDX11L2, they're doing a bad job addressing the functional nature of the protein coding gene that overlaps the so-called centromeric DNA. Their arguments are basically, well, it's spurious. It's not really functional. That's weak. That's been debunked. or they'll say, well, the gene moved over the so-called fusion site. Okay, can we get some empirical evidence for that? You know, so I feel like they really need to do a better job engaging those arguments. And I think as creationists, it would be good for us to do some more research as to why the area is so dense. I think it's dense for functional purposes, and I look forward to researching that some more. Does that help, Brian?
Yeah. Let's continue with our statement.
Donny is arguing with a straw man here.
She obviously rushed to it. She must have watched 3 minutes of it, got triggered, didn't even notice me saying this is data here in this document on the screen that Chris Roop and I.
Just to let everybody know, the reason she speeds up the thing is so she can monetize it. If she wants to monetize it, because she has problem monetizing it if she don't speed up the. That's why the sound. That's why the Donnie's words are sped up there.
We decided to test her argument. We want to see if that's actually true. Because if that was true, that may be 1 point for the evolutionist side.
But you guys didn't test my argument. You tested a different argument. Remember, I searched the fusion site, something that you could check by looking at the screen with your eyes or listening to me literally read out the letters that I searched that aren't a pristine head-to-head telomere-telomere sequence. And just so you can see really what we did, let's scroll up and open up the job details so you can see the actual sequence that I searched. It was a sequence that goes A, G, 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 T, T, A, G, C, T, A, 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 C, C, T, A, A, C, 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 T, A, A. There's our fusion site. You guys tested the latter. These are not the same. The reason...
Okay, she's saying that she tested that. What exactly is she trying to argue for, and is she correct?
Good question. You know, I'm gonna have to share my. Well, I guess you're sharing, so I can't really share.
I can
So
stop
there. Yeah, I think it'd be a good idea just for your audience to see the chromosome 9 data.
Okay, I gotta make it. Oh, you were able to do it. Okay. Oh,
Okay.
Donnie, are you there?
Yes.
Okay.
Oh,
I thought I lost you there for a second.
you can see me now, though.
Yeah.
Or you can hear me. Okay.
Yeah. I thought you froze up there for a second.
Okay, so I just find it so funny how she says the challenge is for us to find the exact fusion site elsewhere in the genome. She wants us to search for the fusion site. So she wants me to take you basically and search the globe for another one of you. Prove to me that there's another Brian out there.
Technically, the burden of proof is on her to prove that it's a fusion site in the first place.
Right. Show us by the evidence that this is a fusion site and why we should find another spot in the genome where this signature exists in its entirety. No, I'm showing you, and this is why I think she's been rather sloppy in her presentation of this challenge, because there's moments where she says, we don't find these sequences, the telomeric calling cards. That's how she says it. We don't find any example or instance where these telomeric calling cards occur in a head-to-head fashion in the genome. That's what she says. And then other times she also throws in the density. Well, if the challenge is also the density, which shows her circular reasoning, then you need to say that every time. But if that's the challenge, it's not really a challenge. It's like, hey, I'm assuming this is a fusion. Now show me another place where we find this exact sequence. Well, you know what? Chromosome 9 right here. This is what Chris Roop and I found. Here's where we found it. You can see the genetic location. You can see how big the chromosome 9 is. And as you work your way down, TTA, GGG, TTA, GGG, TTA, GGG, it occurs many times. And then right here is where the reverse complement sequences, the CCC-TAA connects. So this is pristine. This looks good. This is a great looking site. Here's Erika's so-called fusion site. The telomeric repeats, TTA, GGG, they occur in isolation. They're not connected. Notice the chromosome 9. I've got it highlighted in red. Do you see how connected they are? They occur in tandem. We don't find that at the fusion site. There's a couple. There's one here, and then there's one here that's connected. But for the most part, they're disconnected. They occur independently from each other. it's degenerate. And she has to demonstrate why this 798 base pair region has to be a fusion, in light of the fact that it was never predicted to be this degenerate, in light of the fact that it's pretty weak in terms of the signature, the fact that it's so broken and it's so weak, but also that these elements here, they're found all throughout the genome, and they are associated with DNA function, specifically gene expression. And since the chromosome 2 area, the DDX11L2, that whole region is, firstly, the DDX11L2 gene is linked to, it's co-expressed with other important genes, the expression of other important genes. There's multiple start points for transcription within that region where RNA2 polymerase binds. And so the reason why it's dense could have to do with the function, the activity, if it's a hotbed of genomic activity. But anyways, for the audience sake, this is the chromosome 9 that they say is not a fusion. But yet these occur head to head. Over here, they say it is a fusion. You know, to me, it's arbitrary, it's subjective. And if she says, well, why is it so dense? Well, dense compared to what? Do we have another human genome in another universe to compare it to? No, we don't. So I'll stop my share there, brother.
Right. I had a question, but I can't remember what it was now. But that's okay. I'm gonna continue sharing.
Now, I will say this, Brian, if these areas in chromosome 2 were more chaotic, then I would concede that this is most likely the ancient remnants of a fusion. If they could demonstrate that the DDX11L2 really is just spurious, really is just a pseudogene without any real function, then I would concede that the most parsimonious explanation is fusion. But then I'd go back to the point that says, if there was a fusion, the creation model explains its existence far better. But I feel like there's too many unanswered challenges for the evolutionists. They haven't answered enough of these challenges. For us, we do have to explain the dense nature of the site. But I think the evolutionary community has way more challenges to answer than we do. Does that mean, like, if we were basing this on a scoring system with the fusion, I think the creationists are in the lead. It would be like, 10-1. They have one good argument, the density. But there's so many arguments that we have against the fusion that they're not answering. And they need to stop just repeating already debunked talking points, such as the DDX11L2 is not actually functional. No, they need to engage that in a more sophisticated manner. Go ahead, Brian.
Most of the stuff in this clip is already been addressed by you, I think, because I want to get to the pig argument before we go. So, but I need to click on it real quick.
The fusion.
So I can do that. All right. I'm gonna go to the other clip. All right. Don't mind the big head here. She likes to put in these little snippets just to make us look even more sillier and that sort of thing. It's a question and begging epithet, but in any given case.
Let's hone in on the fusion site itself for the time being. Remember that the head-to-head nature of the fusion site is important, but something that we haven't really talked about is the degenerate nature of the site itself, which is also absolutely critical. Allow me to explain. Remember that chromosomes are capped with telomeres, and the purpose of these telomeres is to protect the chromosome from degradation during replication and to prevent fusion with neighboring chromosomes. These regions are themselves buffered by subtelomeric regions, which look like telomeres sequentially, although they are degraded in comparison. It is in these subtelomeric regions that we also tend to find genes that are members of the DDX11L family, which will be important later. What this means is that in order for a fusion event to occur, the telomeres themselves must be absent or have failed in some way, leaving the subtelomeric regions open to fusion.
Is this correct?
With the pristine head-to-head would imply healthy telomeres. Yeah, and that's why I was looking. So again, in contested bones, and I got to turn this argument into a slide because I think it's spot on. Chris Roop and John Sanford say it might be argued, as she is right now, that since healthy telomeres help prevent end-to-end fusions, that is true. As she pointed out, your telomeres are the end caps of chromosomes, and they help to prevent fusion. And so they say maybe both of the two fusing chromosome tips were severely degraded, as she's arguing here, right, Brian? With both telomeres eroded to near extinction. And so the fusion happens and the signature is weak. And so that's her excuse for why it's weak. And they point out this is feasible, but it is a very weak argument. It seems unlikely that both of the fusing chromosomes were degraded to exactly the same point, yielding a minimized and semi-symmetrical fusion site. And so picture these ape-like chromosomes, now termed 2A and 2B, their telomere sites would have had to been degraded to exactly the same point, coincidentally, to fuse to create this semi-symmetrical fusion, right? And so I think that's a good point that they make, and I'd like to see Erika address it. But I would still go back to the fact that they did not predict this. If I saw documentation of them saying, and the signature of this fusion, if there is one, if we find it, it should be degenerate because XYZ, the answers Erika, the reasons Erika gives. But we don't find that. What we do find is post hoc, ad hoc rationalization, where Erika is invoking this rescue device after the fact, but it wasn't predicted before the fact. Does that make sense, brother?
Yeah, there is something that I was curious about. If this fusion event occurred in one chimp, like, you know, or whatever they think is our evolutionary past here, and it's not occurring in all the other chimps exactly how does, I mean, essentially it would have to be universally passed on in some way, wouldn't it, in order for everybody today to evolve that in their paradigm. I mean, how do they explain that exactly?
And that's the issue that they have. So earlier in my presentation, I asked the question, what if there was a fusion? Well, okay, you have humans here, that's an extant species, chimpanzees here. The evolutionist argues that the chimpanzee is our closest living relative. And so they don't say we evolved from chimps, they say we go back right up here to a common ancestor, about 6 to 10 million years ago. Then there was a split. Humans go this way, or however the camera's mirrored image. Let's say humans go this way, chimpanzees go this way. Okay. They argue that the fusion occurred in the human line after the split. So it's indirect evidence for human evolution anyways, because even they admit that it occurred after the split. Well, problem is, as you pointed out, brother, whoever experienced this fusion, they would be the ancestor of all humans living today, which means every other human lineage at that time would have to be wiped out, with only the lineage having 46 chromosomes surviving for it to be fixed. That is a major problem because that would be a near extinction event. And so it's hard to believe that this genetic blunder actually managed to be passed on rather than that lineage being wiped out through selection.
Does any of them try to argue that it happened like group wise at the same time? Does someone like for example, someone might try to claim that male and female evolved at the same time or something of that nature? Would someone try to claim that like a bunch of them evolved at the same time and fused at the same time? Do they try to use that rescue device?
Good question. I've never seen that argued, because if you had a group of people, let's say you had a tribe of, I don't know, 200 people, the fusion would basically have to happen in a single individual, you know, rather than it occurring in everybody at the same time. Far too big of a coincidence. So that single individual now has 46 chromosomes. Everybody else in the tribe, either that has to become fixed in that tribe, and then every other tribe on the globe or in Africa needs to be wiped out. Either way, that they have to explain somehow through genetic drift and selection. Every single person today has 46 chromosomes and nobody has 48. Well, why did it become fixed and how?
I just realized how implausible that actually sounds, because
Right.
You would have to have it happening not only in a bunch at the same time, but in the same place at the same time. So
Right.
it just doesn't seem
Right.
likely. That's probably
Well,
why they haven't argued that.
Well, and think about this, Brian. Actually, I had a discussion with Erika and we talked about this for about 20 minutes. And I asked her, who experienced the fusion? She said, well, maybe the Australopithecines. Did all the Australopithecines, let's say it was Australopithecus afarensis, did they all exist in unity? Or did they exist in tribes? You know, because humans, historically, they've existed independently from each other. So you have a tribe here, a community here, and they would reproduce within their tribes. Rarely would you get someone from this tribe going and reproducing with someone over here, which means you'd have multiple lineages going on. But if one lineage experiences the fusion, all the other lineages have what, Brian? 48 chromosomes. So they have to be wiped out somehow in order for that one lineage with 46 chromosomes to survive. Very implausible. Very implausible. But according to our model, the creation model, let's say there is a fusion. What do we have at the flood? We have a bottleneck of 8 people. Okay, so the fusion gets passed on into the new world after the flood. We can easily explain how it becomes fixed. So do you see how either way, if there is a fusion or not a fusion, the creation model explains it best?
Yeah. Now, of course, they use that as evidence, but I find it interesting that poses a problem for evolutionists on that, because they claim that chimps and humans are related, but the number of chromosomes is not the same. Now, some people try to accuse us of kind of a double standard, because we can sit there and recognize fusions happening in other animals and other times and so forth. And why not recognize a fusion? But that kind of commits kind of a straw man. It's not that we don't recognize the fact that fusions can happen. Now, telomere fusions, like you said, are rare, but we recognize things like other kinds of fusion events, right?
Right.
All right. All right. Let me continue sharing.
Let me say this real quick as you're sharing, Brian. Either way, the evolutionists, because they assume evolution to demonstrate evolution. So there's no questioning that humans and chimps are related to them. So let's say humans had 48 chromosomes. What would they say, Brian? Oh, look at that. We have the same amount of chromosomes as the chimpanzee. There's evidence for evolution. Turns out we have 46, they have 48. What do they say? Well, there must have been a fusion. And so that's evidence for evolution. So either way, it's up, down, black, white, night, day. It's all evidence for evolution to them.
Why did they choose chromosome, what they call chromosome 2A, chromosome 2B? You said that was chromosome 12 and 13, is that right, in chimps?
Yeah, which they've renamed
2A
and 2B, basically.
Why do they think that those specific chromosomes fuse together?
So basically, and if the audience wants a visual, at the beginning of my presentation, I showed this specific type of technique. It's called karyotyping, where they can visualize the chromosomes during mitosis. And basically the chromosomes are all scrunched up and they can find these banding patterns using this procedure. And it turned out that there was a lot of matches, but then there's a lot of places where there wasn't matches, right? So they cherry pick, but they found matches in some chimpanzee chromosomes with human chromosome 2. And so they thought, well, maybe those similarities in banding patterns has some kind of significance. And then later on, when genetic sequencing technologies became a little more advanced, that's when they were able to sequence the genome. And that's when they discovered what they thought was a fusion, because they had all these telomere repeats, at the so-called site. Does that help to answer, Brian?
Yeah. I'm going to continue.
Sure.
Meaning that if you find telomeric sequences in a pristine condition, even head-to-head, it is likely not a fusion, but the result of duplication events, inversions, and frame shifts. So for the fusion to occur, degradation is necessary on the outset, and on top of that, the degradation may later be exacerbated because tandem repeats are typically unstable. So far, with regard...
Okay, is that the case about the degradation? That degradation is required for the fusion event. Is that correct?
Well, again, she's arguing from the position that says the telomeres would have to be degraded in order for the fusion to take place, since the telomeres are there acting as protectors from a fusion. And so she would argue it has to be degraded to a point where the fusion can take place. But this doesn't have to always be the case, because scientists look to fusions in equids, they look to fusions in other creatures where the signatures are different than what we find in the human chromosome. And if it was that obvious, why didn't they predict it beforehand?
Yeah. It seems like there's a lot of rescue devices involved here. It's like they're coming up. Well, we're going to say this now because we found out this was true, and then you say, well, if this was true, then how come we got this? And says, oh well, this is, it seems like they're coming up with all these kinds of rescue devices to try to help explain it, and then when you attack the truth of the matter, everybody just like accuse you of straw man argument. And it's like, no, it's just the fact that you are using these rescue devices to try to help count for the fact that you don't have any real evidence as it's a fusion site.
Right.
Is that...
And well, I've seen some of them try and argue as well, basically that it was a subtelomeric fusion. And so your telomeres completely broke off. And then it was two subtelomeres, if I'm not mistaken, that fused together. But Dr. Tompkins has pointed out that this would create an even bigger problem, because your subtelomere regions are between, I think, 100,000 and about 300,000 bases in length. And so we'd expect, if that were the case, for there to be even a bigger signature. And so I feel like they're really stuck between a rock and a hard place. And I can almost guarantee that people like Erika really wish that that prediction was out there, was documented before the discovery. If they just said, you know what, we're going to go look for this signature of a fusion and we expect it to be really degraded, rather than them being surprised by the degradation.
Right. All right, let's continue.
To head-to-head sequences located in the human genome, we have human chromosome 2 and human chromosome 9. Human chromosome 2 is degenerate in nature and significantly larger, while human chromosome 9 is pristine and significantly smaller. In fact, the repeats that were found by Donny and group are about 10.
All right, this let me see if I can move this out of my way. I don't know if the little screen is going to move out of everybody's way or not. I've tried moving it before and it always shows up in the same place. But in any given case, I'm moving it out of my way. All right, chromosome 9 fusion signal is 10% size of the fusion site in human chromosome 2. Now, I think she's getting this from an article written by a guy named Glenn Williamson, because Glenn Williamson actually wrote an article on chromosome 2 fusion. It was at least the one I read this morning, and it was a short article, but he said the exact same thing, that and this is an attack, I guess, on the chromosome 2 that we're basically making it look like it's smaller than it actually was. And they're claiming it's 10% size of the fusion site in human chromosome 2. So in other words, it's bigger than what we're saying. Is that correct?
Well, if I'm understanding her argument correctly, she's basically saying that the signal in the chromosome 9 that me and Chris Roop and myself discovered, it is a lot smaller than the signal they find in the chromosome 2. It's only about 10% the size. And so this goes back to the density argument. So it's just her recycling the whole argument from density, which, as we've pointed out, is circular, it's arbitrary, it's very subjective.
Right. Okay, so she isn't talking about the size of the fusion site necessarily, just the amount of the density that's involved.
Yeah. I think in a way they go hand in hand, because you have this fusion site, so-called fusion site in chromosome 2, that is just packed with TTA, GGG, TTA, GGG, CCC, TAA, so on and so forth. Now, they mostly occur independently. They're broken. It's degenerate, but there's a lot more of those sequences than we find in the chromosome 9 area. The chromosome 9 spot that we found, I think, is somewhere between 40 and 45 bases in terms of the length, but the so-called fusion site is 798 base pairs. And so there's definitely a major size difference. That's a uniqueness that the chromosome 2 spot has, but the chromosome 9 is more pristine. So the chromosome 9 also has its own uniqueness. Erika says, oh, the fact that it's pristine means that it couldn't have been a fusion because the fusion site should be degenerate. And again, that wasn't predicted. She's saying, oh, it should be degenerate, but the authors of her own of these chromosome papers are saying, why is it so degenerate? So yeah, there's uniqueness associated with the chromosome 2, sure, but there's also uniqueness associated with the chromosome 9 as well.
In other words, the original paper predicted that they would not be degenerate. You find out that they are degenerate, then you say, oh, well, we expect them to be degenerate. So it winds up being that, like you
right.
said,
Yeah.
hoc ego, post hoc, ad hoc. All right, let's continue.
Percent the size of the total fusion site in human chromosome 2. So far, chromosome 9 is significantly smaller than human chromosome 2, and it has pristine repeats instead of the expected degraded repeats.
Pause that real quick.
Sure.
Notice how she said, you see how they want their cake and eat it too? So if the chromosome 2 area came back pristine like the chromosome 9, they'd be jumping all over that. They'd be like, look at this. We got this pristine site, exactly where we expected. But no, it's degenerate. And evolutionary scientists are asking, why is it so degenerate? They're surprised. But now Erika is saying that this was expected, but the chromosome 9 is pristine. And so that's evidence it's not an ancient fusion. It's really like, as you put it, whatever the data says, they're gonna find a way around it. So now it's expected, but where did they say it was expected prior to its finding?
Right. Yeah, usually when you deal with that, when you're responding to people, it's hard to not respond every few seconds after time.
Right.
Because they make an error every couple of minutes.
Yeah.
By looking at other animals that are proposed to have experienced chromosomal fusions in the same way as human chromosome 2.
I think the pig argument is going to come up.
2. So telomere to telomere fusions. Because we should see the same thing there, right? We should see a degenerate fusion site in the middle of a chromosome that is proposed to be the one that experienced a fusion. So let's take a look. We'll start with suids, so pigs, and the reason we're going to start with them is because young Earth creationists accept them as related animals. They think that all pigs, all members of Suidae, are related to an ancestral group that walked off of Noah's Ark and then diversified into their existing forms. Thereby, if a fusion event occurred in one of these existing species, they have to accept it as a legitimate fusion event.
Okay, just to kind of let everybody know, I don't think Erika fully understands what we believe on kinds anyhow. Again, this echoes that straw man, right? About the us not believing that any fusion event occurred.
Well, and I don't know I don't know how many different pig kinds were brought on the ark.
Right.
She seems to be implying that only 2 pigs diversified into all the swine species we see today. Maybe there were multiple different kinds of pigs brought on board the ark. I mean, we'll typically say with horses there were 2 archetypes, and from those pre-existing DNA differences we get all your different species of horses today.
Now
Same with
Genesis
wolves,
does
but in
say
the case
that
of pigs,
Genesis does say that 2 of every kind was brought on, but it also mentions 7 of
right?
unclean. And there's disputes about what that 7 is. Some people say it's 7 pairs.
Yeah,
Some people say it's 7 exact, but I don't know exactly. I think people
right.
do
And that's
differ
what
on
I'm
that
wondering in the case of the pigs.
because
But even for sake of argument,
pig is considered an unclean animal in the Bible.
yeah. So it wouldn't just have to be 2,
Yeah,
right? It could be more than 2 with varying numbers of chromosomes.
right. What was you fixing to say?
Pardon?
You were fixing to say something?
Well, so this paper here, basically,
Yeah.
recent fusion events.
Yeah.
If you want to continue the audio, and then I'll respond.
Yeah. Let me continue that, but I've actually got the paper pulled up.
Okay,
Um,
perfect. Yeah.
it's not the full paper because you have to pay for the full paper, but it's got the
Abstract.
abstract on it. Yeah.
Yeah.
But do you want me to go ahead and continue it to let her finish the argument?
Yeah, let her finish the argument, because even for sake of argument, we could say there was 2 pigs or 7 pigs or 14 and they all had the same chromosomes, and then there were some fusions. We wouldn't be against that because we are okay with fusions occurring in nature. We agree that they've happened in equids. We agree that they've probably happened in pigs. The question is, did it happen in the human line? That's a whole different question.
Now,
So
they will claim and she claims this as well, a telomere-telomere fusion. So
right.
we'll get to that in a second.
Okay, let's do it, Brian.
Okay.
Chromosome 6 in the modern pig has been proposed to be a fusion of the ancestral chromosome 14 and 6, which appears separate still in the modern Babirusa. This was initially supported by banding, just as Chimp 12 and 13 were to human 2 many moons ago, but further work has confirmed a fusion. Subtelomeric and telomeric repeats at the center of pig chromosome 6. Fascinatingly, they note that the fusion required degeneration in order to happen. So their fusion site looks like human chromosome 2's fusion site, not the signal at human chromosome 9. This is something that effectively all young Earth creationists, at least the ones that subscribe to the Answers in Genesis kinds, have to.
I love the way she said that because it's not answering Genesis kinds, it's the biblical kinds. Where does Answers in Genesis get it? Where does the rest of young Earth creationists get it? We don't get it from Answers in Genesis. We get it from the Bible.
Right.
So I don't know why.
Over and over again.
Yeah. And there were other evolutionists that made this mistake of thinking that somehow young Earth creationists base everything on Kinham and Answers in Genesis and that sort of thing. In the debate with Bill Nye, Bill Nye kept saying in the debate with Kinham that Ham's argument, or not Ham's argument, but Ham's what would he kept saying? He was something about he was making it like the young Earth model position, or like it was originated with Kinham. It didn't originate with Kinham. So it's odd when they say things like that. It makes it sound like that we get all of our position from Answers in Genesis. No, just we and Answers in Genesis are getting it from the Bible. That's where we're getting it from. Biblical kinds are in the scriptures. But anyway,
well said, brother.
I just wanted to point that out because that kind of, when I first heard her say that, that just kind of like
right. Great response. Yeah, she tends to mock answers in Genesis and the idea of kinds. But yeah, I mean, it comes right from the Bible, and that's where we can take that concept from the Bible and make predictions, saying rather than everything being related, there exists separate ancestries with different created kinds, and humans are their own created kind. And here's the evidence for it. And the evidence supports the idea that God created separate kinds, independent from each other. So great points, Brian.
All right, let me continue this.
To accept because it occurred after the diversification within the family Suidae, which they think is a kind of animal that God created and then allowed to diversify. Uh-oh, Donny, your kinds are killing you again. They're getting you. Look out.
Oh my God, Donnie, do you hear that? Our kinds are killing us. Oh no.
I just dodged it.
Yeah. It's like, what the world?
Well, if there were more than just 2 pigs on the ark, with, I'm not an expert at pigs, but I mean, just from these pictures here, we got a pig with horns, pig without. I mean, maybe there were more than just 2 on the ark. Like you said, biblically, they're unclean, right? And so you could have between 7 and 14, and they could have differing numbers of chromosomes. You know, that's something I'd like to look into. But even for sake of argument, I don't think it's an issue, because we agree fusions happen. I'm always careful, Brian. And so you can go back and watch my debates. You can watch my lectures. I don't tend to make universal statements, because the last thing I want is egg on my face. So if I were to say there's never, ever, ever an example of a telomere to telomere fusion, and then a critic shows a paper that says, well, this says there was, then that universal statement I made is kind of coming back on me, right? But I've made the point that telomere to telomere fusions are rare. I've said maybe they've happened, 1 or 2 of them, but they're rare. And in this case, it looks like there is a telomere to telomere fusion. The paper, the abstract, when you pull it up, you'll notice it says this fusion was likely a telomere to telomere fusion. So they're not 100% on it. Yeah, let's see.
all right. Okay, what was that quote from the abstract?
So they say, likewise, we conclude that pig chromosome, about halfway down. Likewise, we conclude that pig chromosome 6 was most likely derived by a telomere to telomere fusion. And so the reason they say most likely is most likely because this is something that would be historical. It's not like they observed this fusion in nature or in the lab or in a species they're studying in real time. They have looked at banding patterns, as Erika explained, and they have theorized that this chromosome is due to a fusion, and specifically a telomere to telomere fusion. But the paper itself, which is, Erika's kind of vague here. She makes assertions and claims, but she doesn't show much from the paper, because she's saying, oh, it was degenerated in the same way that the human chromosome was. Okay, well, the human chromosome too is 798 base pairs. There's only about 9 or so TTA GGGs intact. There's about 43 CCC TAAs intact, the reverse complement. We have documentation of this. I can show you a visualization of the area. I can show you a visualizing of the chromosome 9 area. But I watched her video a couple times. I didn't see any details presented on how degenerate it is. I didn't see her show us the site. I didn't see a breakdown on how many instances of TTA GGG we find. And so she's kind of just asserting that it's degenerate. But I'd like to compare the site to the human chromosome 2 site, rather than just take her word for it.
Right. Do you find her arguments a bit circular here?
Well, I do. It's kind of apples and oranges. I mean, we got pigs, right? And then we got humans. Well, God created 2 people, Adam and Eve, 6,000 years ago. But when it comes to animals, God created populations of animals. He didn't just create 2 pigs, 2 cats, 2 dogs. He created animals in fully functioning and working ecosystems and populations. Yes, we have a bottleneck at the flood, but in the case of pigs, you could have 7 to 14 pigs with varying degrees of chromosomes because fusions could have occurred in the pre-flood world and been passed on. So it's kind of apples and oranges the way she's trying to compare pigs to humans. But I also have no choice but to consider this argument for now as being an assertion, because she didn't really show enough details for us to make any real valid conclusions. She didn't show the details of the fusion site in the pig chromosome 6 like we can do with the human chromosome 2. I mean, the human chromosome 2, we know exactly how many bases there are. We know exactly how big it is. We know exactly how many repeats there are. We have a lot of information on it, but I just don't see her presenting that kind of information here with the pig. What's her idea of degenerate? I want to see the site, you know?
Right. She seems to assume that if this is an assumed telomere-telomere fusion, then the evidence must be the same for that fusion site.
So I guess she's arguing that I have no problems with this being a telomere to telomere fusion. It doesn't change the fact that they're rare in nature. I find it curious that in all of the searching of the public database within these papers, you only find 1, maybe 2 examples of a telomere to telomere fusion. So it is very, very rare.
I do have a question that was on my mind earlier. I forgot to ask it. But what do evolutionists? You said that a lot of evolutionists are not up to date on the papers and stuff out there. What do evolutionists in the peer review papers say concerning that fusion? Do they think the fusion occur? Are they divided on it? They argued for different things, and there's kind of contradictory models out there. I mean, exactly what do evolutionists say within the literature itself concerning that?
Within the literature itself, they are basically in universal agreement that human chromosome 2 is the result of a chromosome fusion. And that's why they've even renamed the chromosomes in chimpanzees 2A and 2B. That's how sure they are. But it's because they don't really have a choice. Humans have 46, apes have 48. How do you explain that? Well, the most parsimonious explanation, if you hold a universal ancestry, would be a fusion. But as far as I know, and from my study, they're not as confident as Erika that the degeneration should be expected. Or because I've shown the quote from the one fusion paper in 2002, where they actually express surprise over the degenerate nature of it. So I think that's the difference. I don't think they're as confident as somebody like Erika is. Although they believe it happened, I don't believe they're as confident with Erika that the nature of it, the signal, is exactly what was expected.
All right. Let's go and continue. Do we need this paper anymore or not?
Not really. I mean, you can basically see that they're not 100% certain that it was a telomere to telomere fusion. They're inferring, but you know, I don't have a problem with it being one.
Part of the reason it's inferring it, they do say that there's a telomere centromere fusion of 2 ancestral chromosomes, but that's inferred too, isn't it?
Yeah, because much like the human chromosome 2, they're not actually observing. Like, we can observe fusions in real time. We see fusions in cancer cells. Those are telomere to telomere, but those aren't good. They're not going to be passed on. And so they're just examining the genomes of these organisms, pigs, for example, and they're looking at the banding patterns, as Erika explained, and they're inferring a fusion. And it looks like there probably was a fusion, and they're concluding it's a telomere to telomere fusion. Erika is going to say the signal of this fusion is very similar to the signal found in human chromosome 2, but she doesn't really provide those details. She doesn't show us the site in the pig chromosome, does she? I don't think she does. No, she doesn't.
Let's go ahead and let her continue that argument, and
Sure.
we'll get one response to it because I do want to try to wrap this up. It's getting kind of late here. It's probably getting real late your place because it's you're an
Yes,
hour ahead of me.
my voice is starting to go on me, I can tell.
All right, let's go
But it's
ahead and
worth
let
it,
her
brother.
finish.
It's worth it.
Let's go ahead and finish her argument, then we'll respond to that, and then we'll close it out.
Cool.
So under the kind system, younger creationists do accept that telomere-to-telomere fusions happen and that these fusions require degradation, resulting in non-pristine fusion sites. This work has stood the test of time, and more types of fusions have been discovered within the Suidae family. So we have established that telomere-to-telomere fusions do happen and that degradation is necessary for them to happen. Thereby, degradation characterizes fusion sites.
We noticed that second side of her argument. The first side was okay because we know fusions happened, but it's kind of strawmanning us because we don't claim that it doesn't happen. But then the second half of her argument then claims that degradation is necessary for fusion to occur, and you would argue that that's not it's not necessary.
And then she has to answer the question, why did both chromosomes degrade to exactly the same point to create this semi-symmetrical signal? I'd like to see an answer to that.
Right.
But what is actually going on in human chromosome 9? It's a pristine head-to-head sequence of telomere repeats. Can conventional science explain it? We will now consider the equids, specifically horses and zebras, since younger...
All right, she's going to go into horses and stuff. We're going to go in and not do that unless you want to, because I mean, basically, it's going to cover a lot of the stuff that we already talked about. Is that okay?
Yeah, I think. Like, if she responds, we could also do a part 2. I'd be happy to do a part 2 with you if you wanted in the future.
I wouldn't mind actually to do an actual full response video to that particular response at some point later point with you. And then we kind of can go over those specifics and be able to cover more ground and have more time to do that. But we covered a lot of things in this video, but I did want to give you a chance to respond to a few things she said in there. A lot of the other stuff that she said, you've already responded to already anyhow. So how would you summarize when it comes to the evidence, when it comes to the issues and the arguments on this? Summarize your case and identify how this reflects on the case that they make in favor of this position, and then we'll close it out from there.
Yeah, I think, firstly, if for sake of argument we were to agree that there is a fusion, I pointed out in my presentation that it would be considered an apomorphy or apomorphy, where it's unique to one lineage. It's a trait unique to just humans. And which model best explains the presence of a fusion? I would argue that the creation model, since we have 2 bottlenecks. But for the evolutionary model, this would be a near extinction event in order for every single person today to have 46. And nobody has 48. So that person who experienced it would have to be the ancestor of all people today. And so I'd like to see from the evolutionists like Erika, rather than just a story on how this could happen, how this can be fixed, how all these other lineages can be wiped out, I want to see a mathematical model. I want to see some empirical science. If they're so confident that this happened, the question is how, and also how is it passed on? Secondly, I'd like to see a more sophisticated approach to addressing the functional nature of both the protein coding gene that overlaps the so-called centromeric DNA and also the DDX11L2 long non-coding RNA gene that overlaps the telomeric DNA. Because I would argue that the evidence at this point suggests that these functional DNA elements within both genes are necessary for the genes itself. It's basically functional units of DNA within healthy human genes. And their argument is, well, the DDX11L2 gene, it's a pseudogene, it's not really functional. And I've addressed all the arguments that they've made. as to why they believe it's not really functional. I'd like to see them counter those. Same thing goes for the protein coding gene that essentially overlaps the alphoid repeats. And we find these telomeric repeats, TTA, GGG, CCC, TAA. We find them all throughout the genome. And we even find instances of very pristine cases, like in chromosome 9. And nobody would say that those are the result of fusions. And to me, that says the chromosome 2 area doesn't have to be the result of a fusion either. They're going to ask about the density, and we can have an entire debate itself on why that region is so dense. But I would argue that their assertion that it's dense because of an ancient fusion, it's circular, it's arbitrary, and it's very subjective. And so that's my summary, brother.
Awesome. There's a lot of speculation involved. You mentioned that they just kind of invent a story. And that's typical of the evolutionists, and a lot of the evolutionists that responded to me, a lot of stuff they're saying is conjecturalized. So they're no stranger to conjecture.
Amen. Well, no, they are. They're masters at it.
Right. It has been great. It's been fun. Thank you for joining me on here, Donny. I know it took us about a month ahead of time to try to schedule this up, but that's okay. But it's been great. It's been fun. I loved it. I enjoyed it. I learned a lot. Hopefully everybody watching this has learned a lot. I'm sure Erika's going to make a response video to this.
I'm sure.
You know, I was just thinking just a second ago when I made the comment about how she speeds up the time to, you know, because money reason or because she likes to monetize her videos. I realized that my words might actually be sped up when she said they're watching it, responding to it. So that's gonna be kind of funny. But anyway, I do plan on making a response video to the response video she made against me and Tompkins. It's going to be the biggest response video I ever did on my channel. I'm going to make it part of the Genesis series. I'm inviting all kinds of guests to it to you know at different points, and I'm kind of going to edit it and put everything together, and so it's going to be a great discussion. I already proved her wrong about what she said about Richard Buggs, so. Because she claimed that Richard Buggs took a 96%. No, he doesn't. Tompkins was right. His last study of it was in 2018, and he took the 84.4% number. Right now, he doesn't want to commit because he doesn't know. He hasn't studied it since then. But he said that I had an email exchange with him actually because I was going to invite him on my channel for an interview over that just to sit there and see what he says because Erika claimed that he had a different position than other than the one that he's made public, and but in any given case, he does take he took the 84% number in 2018. Now he's not committing to a number yet because he doesn't know if the chimp genome has been more sequenced, but we know that it hasn't because he was using Pantro 6 in 2018 when he redid this number figures. And so he actually took an 84.4% number. That was the maximum, he said. He said that at that point, since the rest of the chimp genome had not been yet sequenced, 95% at best was a prediction. Now, that was his words. So awesome. Erika was completely off track. Completely off track.
Okay, so Richard Buggs did not, in fact, change his mind on the number. He's still going with the lower number.
Well, yeah, he did in 2018, which was, according to him, was the last time he did the study on it. Awesome. And right now he's just kind of like, I don't know whether it's been sequenced or not, and so he doesn't know what the numbers would be today. But of course, he says in the blog that he was using Pantro 6, which was the most recent in 2018. That's the one that Tompkins used in his most recent study. In the 18,000 contigs that he examined. And so we know that was the most recent chimp genome that was sequenced. I think there's like 5% left that they haven't sequenced yet. But he said that if they do fully sequence it, he said he was going to do a new study and revisit those numbers again to see where they fall. But I know that in 2018 in that blog, and he in fact even linked the same blog to me, that if I want to know what his position is, that was it. So that was that. But it was a very cool conversation. I plan on, when I do the response video, I plan on showing the email exchange that me and Bugs had. I plan on showing the post that he put on bio logos. He has a series of discussions on that. He talks about it. In fact, in one place, and it was his longer post when he was responding to like Swami Das and others, he said that in all the criticisms he had since he put that number out, nobody ever challenged whether it was scientific or not. Nobody said it was unscientific what he was doing. And he said that. He said if they did, then he would have sat there and revised those numbers. He would have changed it if his approach was unscientific. So I thought that was pretty cool. But anyway, I've got all that down. That's one of the reasons why I got several tabs on there. But I've got several tabs since putting that on there. But I don't want to take them off right now because I do plan on putting that into my video. So she was just factually inaccurate, and there's areas that I already did on disproving her on what she said on Australopithecines and things like that. So it's a cool. It's going to be a really, really good discussion. But a lot of these people,
I'm looking forward to that, Brian. That's going to be awesome. I'm looking forward to that.
I'm gonna really focus on it probably after this video. I want to focus on it. Dr. Lau approved doing an interview with me over the math argument, where she tried to argue from weighted averages and things like that.
Right.
Now, of course, the way they did it, it was a little weird to me because all the numbers came from the CBC files, including the longer sequences. But in the shorter sequences is what she averaged out. And because it comes to 84.38% or something like that, she says that must be how Tompkins did it. Now, there's nothing that in those papers that said he did it this way. She's assuming he did it that way because of the 84.38% that it comes to. But the longer sequences was also part of the amount of data that he was looking at in the paper. So I don't understand why wouldn't you sequence that out too with the shorter sequences? I don't know. Maybe I can ask Dr. Lau if he comes on here. But anyway, Dr. Lau's like really busy. He proved it, but they got like lots and lots of other interviews and stuff like that. And I talked to his assistant actually, might be yesterday. I talked to her, and she told me that they were just swamped right now, and it could be a couple months, maybe longer. I don't know if that's going to happen or not happen, but I do know that it took me almost a year before Dr. Lau came on for my distant starlight video, and that was the highest viewed video on my channel. So
yeah, you've got a couple thousand views on that one, I noticed.
Yes, definitely. It's over 2,000 views now. So it is the highest viewed video on my channel at the moment. And so it was well worth the wait. So maybe it'd be worth waiting a few months or so for that interview to happen. But it's only going to be like 15-20 minutes because I'm kind of putting it together with a much larger video. So it's just getting Dr. Lau in to respond to one specific argument, because in connection with him being an astrophysicist, he's also a mathematician. In his PhD, he actually has a minor in mathematics. So he's actually would be considered a mathematician as well. And he uses a lot of math in physics too. So he's like a expert when it comes to mathematics. And I thought I could get him on here and have a discussion and look at her specific argument on that. But anyway, you look like you're about to go to sleep. So
no, I'm just excited for this upcoming show. I think that's awesome. I like that, you are very similar to me in your approach, that you are willing to engage the critics. If they respond through a video like Erika did, you're ready to battle her arguments. You're ready to take them on. So I'm looking forward to that, brother. That's awesome. This has been a blast. I think this has been really good. 3 hours has flown by, and I'm glad that we were as comprehensive as we were. And sometimes I can be admittedly a little long-winded, and I'll tell you why. It's because I know these critics will typically respond. And so rather than just giving a very short answer, I like giving answers that anticipate a lot of what they would say if they were to criticize it. It's like, killing two birds, one stone. And so I know that they're going to keep focusing on the density, and they're going to keep being circular and being guilty of these logical fallacies. But I'm glad that we were so detailed
And
tonight,
bring up
brother,
rescue
in
devices.
addressing those.
And also, you're talking to someone who is long-winded. I get accused of that all the time when I respond to people. Sometimes I have to split my post up in multiple posts because because it's too much in one given post. And some people don't even want to read the post, but I can't help it. It's easier to level accusations, including conjectural ones, at someone than it is to respond and explain things and have to back things up. And of course, I like backing things up. I don't like just saying, "Hey, this is true. Take my word for it."
Right.
I mean,
Well,
I don't
and I
take their
find
word for
that
it.
I find that's what Erika did a lot with this pig example. I want to see the pig site and then the human chromosome 2 site, and let's compare them, apples to apples, and not just take her word for it. And what I like to say oftentimes, Brian, is these critics, they say more things wrong they say more things wrong in a sentence than words that are in that sentence. And so it takes us a lot to break down what they're saying and refute it.
Right. All right. It's been great, Donnie. I really appreciate you on here. It's getting really, really late. Do you want to add any closing thoughts to anyone before you leave?
No, I think I summarized it all a little earlier, and I'm happy with that. If people want to, I've got them behind me. So I guess the last thing I'll say, if people want, a detailed breakdown on all of this, I highly recommend Special Creation. This is about 300 pages. I have a whole section on chromosome 2 in this. And then for ERV, since I really only covered it for maybe 10 minutes at the end, I've got a book here, Endogenous Retrovirus Handbook. This is a couple hundred pages, but it leaves no stone unturned on the topic of ERVs. And all of my arguments are backed up by the secular literature. And so, yeah,
Hey,
I appreciate you doing this, and I think this has been great, brother.
no problem. Hey, I was going to show you something. I forgot.
You got it.
Yeah, I was going to show you that I got that Apes as Ancestors. I got it for free.
Wow.
Bergman sent it to
I
me.
want to get myself a copy of that. I'm going to have to ask Jerry next time I talk to him.
Right. But he said that they're revising it is why it's unavailable now on Amazon and stuff. But he told me he would send me a copy, and he did. He did. I sent him my address, my email, and he sent me a copy. So I appreciate that. If Dr. Jerry Bergman is watching this, thank you again. I already sent him an email thanking him, but that is great. That is awesome. I enjoy it. I'm been reading it since then. But any good case. All right. By the way, what do you think of my last video on with Jerry Bergman?
I thought it was fantastic. I watched the whole thing. And I may have a few minutes left, but I know I watched most of it. His presentation was great. He gave a similar presentation on mine. And I really liked, as usual, brother, I like how interactive you are. And so I appreciate how you pulled up Littlefoot, you pulled up Lucy and all the different bones, and you allowed Jerry to address each point. It was a systematic takedown of human evolution, put it that way. And so that was great. You asked some great questions. Jerry had some excellent answers. I really enjoyed that. I'd recommend everybody check that video
Right.
out.
And the funny is, you know how evolutionists responded? By attacking Dr. Jerry Bergman directly without dealing with his arguments. I kept saying, why don't you deal with his arguments?
Ad homs.
Right.
That's always a sign that they're losing the debate.
Right. One guy, a couple of them actually got the claim that he attended this one school. They kicked him out and they got shut down. And then when I looked it up, I found out there was only one that claimed that. And it was Rational Wiki, which has evolutionary assumptions and anti-young Earth creationist arguments all through it. Are you familiar with Rational Wiki?
Yes. Oh, yeah.
And it's but every other bio, including his own bio, said nothing about attending that school, said nothing about getting a degree from that particular school, said nothing about, any of that. And the schools that he did is on his bio are credited. So that was it was a foolhardy guilt by association type argument.
Right.
But and it wasn't even a good one because it was false information
It's
and
a
cherry
sign that they're
picking,
losing the debate.
right?
It's a
And
sign that
cherry
they
picking,
can't actually deal with the arguments.
and it was cherry picking because when you look it up on Google, because one guy said he did a quick search. Well, when you look it up on Google, you find a bunch of regular bios as well, including on creation.com, which is CMI's website. You got one on Dr. Jerry Bergman's website. I forgot what it was, but it's basically one that has lots. I think it's Evo Creation or something like that, but it's got like a bunch of articles and stuff on that website. But you can find his bio on there as well, and you got like a bunch of different things that showed the bio of it, and it's none of them agreed with Rational Wiki. So it was a really cherry picked type argument. But anyway, but that's how they responded. So that kind of impresses me that they can't deal with the arguments, so they just attack the person.
That's a good sign that you did a good job with that interview, brother.
Awesome. But anyway, okay, we're gonna go ahead and close this out. And thank everybody for watching, and God bless. God bless.
God bless.
Thank you for joining me on Apologetics 101 for this incredible interview with Donny Bodensky from Standing for Truth Ministries on alleged chromosome 2 fusion. And please subscribe to Donny's channel if you haven't subscribed yet to Donny's channel. Donny's channel is really good. He does a lot more content on his channel than I do on mine. And because he has debates and he also has interviews and lectures and things like that on his channel. He does a lot of stuff on his channel, and he covers a lot more topic matters than I do as well, including things like on eschatology. I don't cover anything on eschatology on this channel, at least ever right now. I thought about having a prophecy series eventually. So right now I'm not doing anything on eschatology and so forth. So he's got a really good channel, and he does a lot of stuff on there. So please subscribe to Donny's channel if you're not. I will be linking his channel as well as his website in my description box below. So please check those things out. And I might also think about linking certain articles that he's written on the subject matter of chromosome 2, including responding to Erika from Gutsick Gibbon. I think that would be helpful too, and I'm going to link those in the description box below as well. And I hope you learned a lot from this. I think that the evidence against a fusion event happening is way too strong, and the claims of fusion happening is just way too weak. You got the fusion site, alleged fusion site alone with 798 bases. Where telomeres typically are anywhere between, like Donny said, between 5 and 15,000 base pairs, and you'd have to assume all kinds of speculations. Maybe they lost a lot of information there. You would think it would be bigger even then, but and Donny pointed all this out, and you got issues where you have the same head-to-head sequences, other places in the genome, so it doesn't really match very well. The density argument appears to be a bit circular, and I just don't think that it matches very well. But we do accept that fusions can happen. There's always that possibility. As Donny pointed out, it seems to be much more plausible that if a fusion event happened, it's much more accountable within the young Earth paradigm than it is within the evolutionary worldview. Because you'd have to assume an extinction level event, as he put it. You'd have to assume that, because otherwise you'd have to have the fusion happening, universally among that branch, that branched out from those chimps in the evolutionary paradigm. So I think the evidence against a fusion happening is strong, but if it did happen, it would be much more accountable in the young Earth paradigm than it would be the evolutionary one. It wouldn't necessarily prove that humans and apes are related, which is exactly what the evolutionists believe, in which case that's what they're trying to support, because chimps have 48 chromosomes and we have 46, and that presents a problem for the evolutionary model, I guess. In any given case, I hope you liked this interview, this discussion that we had with Donny. We covered a lot of ground. We responded to some of the arguments of Erika from Gutsick Gibbon. So it's been incredible. We have a lot of content that was there. I loved it. I enjoyed it. And don't forget to like the video, give the video a thumbs up. So that way this video can get out to as many people as we can possibly get it out to. Don't forget to subscribe to my channel. I've got lots of videos in store. Don't forget to hit that notification bell either. So that way you can be reminded by YouTube every time I produce a video. I've got lots of other videos planned up. I do plan on doing a major response video to Erika from Gutsick Gibbon, and I hope you join me on for that, because it's going to be epic. And I plan on making it part of the Genesis series, because it's going to be piggybacking off of the Tompkins interview that I had with Dr. Jeffrey Tompkins. So it's going to be a major a response video. Will probably be the biggest response video I've ever had on this channel, and I'm going to be making it part of the Genesis series. So be the only response video, or at least the first one. Hopefully it'll be the only one, because I usually don't make response videos part of the Genesis series, but this one, even if it's related to Genesis, but this one I'm gonna make part of the Genesis series, because it actually responds to a lot of critics' arguments, and I'm gonna be bringing together lots of different components to this discussion. So please don't forget to subscribe to my channel. Don't forget to hit that notification bell to be alerted to all future videos, and that's it for today, and I will see you on the next one. God bless.